Hello, everyone. Good morning, good afternoon, good evening. Welcome to this webinar organized by Humane Society International. We are going to wait a few minutes just to allow everyone to join and to participate before providing some initial welcome remarks. Okay, I believe that's in the interest of time, since we have a very tight schedule for our webinar. So welcome to the webinar dedicated to the transition to non-animal-based vaccine batch release testing, focusing on trying to provide some important information about policy and regulation theoretical aspects and case studies for this transition. Very few information for you all joining this webinar. You will be unmuted and without video to facilitate the flow of the webinar in case of slow connections. We would kindly ask you to put your question into the Q&A box. We will try to reply to all the questions as much as possible. If it's not possible, we will try to answer them in written inside the Q&A box. The chat of this webinar is not monitored, so please do not put your questions there because we might miss them. So please use the Q&A box. What is also important for me to remind you that this is the first webinar of a lot of online webinars and workshops. So please stay tuned and we will be happy to invite you for additional discussions and conversation during this year. So for today's, as I said, so we would like really to provide you with some both theoretical aspects and concrete case studies with regards to the transition from in vivo to in vitro testing for human vaccines, batch release testing. And every presentation will be followed by a short Q&A, and I will introduce the speakers at the end of every Q&A session. This webinar is part of the Humane Society International project that is called Implementation Plans for the Replacement of Animal Testing for Human Vaccines. This is a project that is sponsored by the Bill and Melinda Gates Foundation and wishes to accelerate the implementation and the regulatory acceptance of non-animal approaches for human vaccines batch release testing. We would like to support all the stakeholders that are interested to understand how to implement those methodologies and how this implementation can work within the next 5 to 10 years time. So we will have a lot of opportunities to collaborate with many stakeholders individually and collectively through webinars and workshops. And why this project is happening right now, because within the last 10 years, we have seen a lot of important projects and transformations in terms of development of those methodology, but at the same time, the creation of a very strong scientific community of experts that have been working to support this transition. At the same time, we have been seeing how many regulatory agencies, national control laboratories, and pharmacopoeias have been started to use those methodologies, accept those methodologies, and have a positive dialogue with the stakeholders that are interested in this type of transition. If it's okay with you, we would like also to learn a little bit from about you, why you are here today. So if it's okay with you, can you please go to the slido.com or just take a picture of the QR code to answer a few questions. So give me 1 second, that's we go to Slido in a second. Okay, thank you. We would like to know from where you come from. Thank you. We see that we have a nice representation. Please do continue to vote. If you have not yet done it, we also would like to learn from which organization you are from. So if you are working within a national regulatory authority, a national control laboratory, or from a local pharmacopoeia, from a public or private organization. Please do continue to vote also on the other questions that we are very curious to understand from you all. It will be very important also when we get to the Q&A session to understand how we can set up in the best way possible the questions and the answers. Thank you. Just please do continue. What is also important for us, it's about what are your expectations from today's webinars. If you can just choose one keyword, it will be again important for our speakers to set up the expectations. Thank you. That's great. Please do continue to work on your answers. That's great. Thank you very much. I quickly go back to some final remarks about my presentation. What we would like to do through the projects that we are running within Humane Society International is really to secure the opportunity to have you all engaged in all the future activities and allow you really to get into the dialogue about what are the concrete opportunities in terms of developing the methods that still need to be developed, and also implement some of the methodology that are already available, and some of the organizations around the world have been already successful in implementing it. With this, I thank you very much for being with us since the start of this webinar. I leave the floor to our first speaker that is really going to set the scene for this event. It's Dr. Dean Smith from Health Canada that is going to give you an update with regards to what is the status from a theoretical aspect about the substitution of in vitro to the in vitro technologies for human vaccines. Thank you, Dean, for being with us. The floor is yours.
So thank you, Laura, and thank you to the organizers and the funders of this to provide an opportunity for us to share our our thoughts. I'll just bring up my slides here. Switch that to center. Great. Okay, so thanks again. So this is a theoretical, practical, and actually more philosophical presentation on substituting in vitro for in vivo potency and safety assays, science versus the fear factor. And I want to credit my colleague Laura Mallet with EDQM for suggesting the punchy last part of that title. This is a very much collaborative project coming out of EDQM and Group 15 and Group 15V. You see the members of the working group here and also colleagues at the bottom who were either not quite part of the group, but very significant contributors to this. I want to shout out to Emmanuel Chantrin, who recognized the voices within Group 15 who were looking for a new path forward for changing from in vivo to in vitro assays, and challenged us in Group 15 to come up with a plan. It, as you see, took quite a while to do so. And what the main point of this talk is going to be the changing of hearts and minds, because all of us have, substantial backgrounds in the areas we're responsible for within our regulatory functions or industry functions. But all of us come with our own sets of ideas, and this project is all about changing ideas. And one of the name I want to shout out here, there's quite a few of the speakers you'll see on this group that are presenting today, and their names will come up over the day. I want to put a shout out to Jerry Calver, who's at the bottom on the Health Canada list. And when Maria Baccastrada and myself joined Health Canada back in 2002, Jerry Calver had been there for some time already and was already at that time had discontinued the reversibility test for histamine sensitization for pertussis toxin vaccines, and that was long before any of these projects started. So he's become something of an inspiration for us in this space. So overview is to identify the barriers to the transition from in vivo to in vitro assays for key legacy public health vaccines. And this is the problem here is the beliefs that we have about this and the negative feedback loop that creates. So if regulators believe that in vivo assays are essential to maintain the safety of vaccines, and they are perceived, by industry as non-supportive of change, then industry will not invest in innovation and new assays, and no change can occur. So what we've learned through Group 15 and the 5214 exercises, the path forward is a more science-based, less fear-driven, more risk-aware mindset at European Pharmacopoeia for the Group 15. And of course, we've all been science-minded for some time, but really challenging our own personal beliefs to go forward. And so the key message here is that the dilemma related to in vivo assays and resolving this legacy was this science thinking within the industry and within regulators to develop stories that better reflect the reality to circumvent this negative feedback loop. So I can't hear or see you all now, but depending on the audience, this slide often gets a bit of a laugh, especially if there's a large number of industry folks in the audience. However, this is an example, this 5214 project is an example where regulators have stepped up to create a new path forward for these transitions because of their new stories they have in their mind. And this is that larger story about that, how that happened at Group 15. So what is the goal? What are the hurdles? We all have a common goal, if you will, is make sure that vaccines are of sufficient quality, safety, and efficacy. And the other side of that balance tip board here is that the availability of these manufactured products. So unless we have appropriate assays in place that are reliable, that don't require multiple tests and can get products out the door without a whole lot of controversy, we're not going to meet that. We may have highly effective vaccines that we always have, even with in vivo assays, but if we can't move them quickly to market and move them in an efficient way to multiple markets worldwide, we're going to have some serious problems. So the solution to this requires innovative thinking by both regulators and industry, and science-based decision-making to support manufacturing, testing strategies that can lead to global regulatory harmonization and convergence. This convergence and harmonization globally is extremely important because even if we have changes that occur in one region of the world, for example, in Europe and quick movement to more appropriate in vitro assays. If those requirements for animal assays still exist in other parts of the world, that means manufacturers now have double the task or triple the task to do assays that match different jurisdictions. So harmonization is another key initiative here. And 5214 provides a framework for perhaps a more rapid change through a science-based approach, supporting also ethical considerations. So the idea of having in vitro assays is not a new concept at all in vaccines. We've had many vaccines on the markets for some years that don't involve any in vivo assays. So this is not a exactly groundbreaking idea. Examples here include the human papillomaviruses, HPV, which have been in the market for many years now. They are recombinant viral-like particles plus an adjuvant controlled with physicochemical methods and ELISA, again, no in vivo assay. Or another example is meningococcal, pneumococcal conjugate vaccines, which are defined polysaccharides plus carrier proteins, controlled again through physicochemical methods, essentially. And again, more this rather horrific experience we've had with the pandemic, but for vaccines within EMA, authorizations and North American authorization for COVID-19 vaccines, these were all exclusively through in vitro quality control methods, some of them being almost exclusively physicochemical, but with some in vitro cell assays as well. And that was regardless of platform, whether that was mRNA, subunit, or viral vector. And there were animals involved. Of course, we went through a great number of animals during the pandemic, all under humane conditions, of course, within those specific jurisdictions. But they were for proof of concept and preclinical studies, not for the quality control. So the key message here is modern quality control strategies for vaccines involve a combination of physicochemical methods, in vitro methods to monitor critical quality attributes, to maintain the efficacy and safety profile of products that were established at the time of licensure. So we have over decades moved to a different view of this, and it's of course still evolving, where quality is built into the process under CGMP that involves the design, the development, and process controls and manufacturing. We have consistency of monitoring throughout the manufacturing process, and for some multivalent conjugate vaccines, you may be testing at 300 intervals throughout the manufacturing process, which involves starting materials, fermentation, purification, and activation, formulation, and finally a release test. And certainly within the next 10 years, we may even see the disappearance of release testing. It'll be solely based on in-process controls of a process, and it will then go out the door. So also note that for approved rabies vaccines in North America and Europe for decades have been formulated based on in vitro assays. And the formulation assay is a critical point, of course, in a vaccine, especially for, a rabies vaccine, by the way, is a life and death vaccine. If you are challenged with rabies virus and you do not have an appropriate vaccine on board in terms of an immunization, you are going to die. And it's really that simple. And yet those products have been manufactured using in vitro assays for decades, and then they must go through the dreaded National Institute of Health animal rabies potency assay, which is a bit of a roll of the dice. The variability in that assay is between 25 and 400%. It's massively variable and very time consuming. But yet those products have been really controlled properly by in vitro assays. Another example is, again, these conjugate vaccines, which are issued sort of in micrograms per milligram, a defined component, for example, polysaccharide or adjuvant, and those are exclusively controlled by physicochemical methods. So this idea has been around for a long time, and we're now talking about how we get them more rapidly. The trouble is, though, that in spite of those possibilities, we have many years, decades, in fact, of stagnation. And one of the key examples that drove the 5214 project for many of us in Group 15 was the rabies NIH test, which at that time, which we started in 2012, as mentioned, was sort of 45 or 50 years in the making for trying to transition, and we'll explain that story a little better later on. And various methodologies have been used: single radial diffusion assays, the SRID, in vitro neutralization, and then glycoprotein ELISAs. But through these one-to-one comparisons, failed many times. And specific regulators I knew and respected, very intelligent people, would just simply not let go of that assay on the belief that that was a critical method to protect the potency and safety of those products. Similarly, the abnormal toxicity test or general safety test persisted for many, many years without change, and we'll tell stories about that a bit later. The histamine sensitization test for pertussis vaccines, again, 20 years of stagnation at the time we started this, and the same too for toxoid reversibility, potency, and safety assays for DPT vaccines, and then the persistence of rabbit pyrogenicity as a control for pyrogens as opposed to alternatives like the monocyte activation test. So the key lesson here is this, what we need to get out of this is as a scientific, less animal-centric mindset was needed at an international level to implement alternate in vitro assays required for world markets and vaccine manufacturers. So what are the key limitations of in vivo assays and considerations for in vitro alternatives? These are paraphrases from European Pharmacopoeia 5214, this general chapter. So these decades of inactivity in this area, not total inactivity, but greatly slow progress, what it has to do with the variabilities in vivo assays and multiple failures in multi-center international collaborative studies requiring one-to-one comparisons. And again, a key one is the rabies NIH test, which went through multiple studies, which could not get through the comparative, not because of the failure of the new method, but because of the variability of the in vivo assay. So this was a major blockade. Also, most in vivo assays predate ICH-Q2R1 or VICH-GL2 guidelines, yet these in vivo assays are considered validated because they are compendial, hence one-to-one comparisons are challenging or not even possible in some cases because precision, reproducibility, limits of detection, etc., which are standard ICH requirements, are not available for the in vivo method and would be unethical under certain EU constraints to suddenly reproduce those. And by the way, I must point out that while Europe, for example, had 3Rs constraints, requirements, in fact, to replace in vivo methods with suitable in vitro methods, that there still had that not overcome the barriers to that, which is why 5214 was developed. So again, this deconstruction of myths, while properly established in vivo methods have the potential to measure complex functional responses for demonstrating proof of concept, they do not predict the responses in the target population. They are merely highly variable bioassays. And this is a really key idea. We're not gaining much precision at all. We're losing it, and we can control things through alternate means. In vitro alternative assays, QC strategies using one or more new methods will likely assess the same quality attributes differently. Hence, the expectation of a single agreement between two in vivo and in vitro methods is strictly not scientifically justified. Yet the in vitro test strategy can and must provide at least the same confidence regarding the control of the key quality attributes, and the case studies you'll see today will support that. So the new approach with 5214 essentially is that replacement, this one-to-one comparison, is a standard model we all understand quite well, which again, for these highly variable assays was problematic. What we're proposing in 5214, or what is now established, under 5214, is to facilitate the implementation of in vitro methods. And we're talking about appropriate in vitro methods, of course, which are sufficiently stability indicating, as substitutes for existing methods in cases where one-to-one assay comparison is not appropriate for reasons unrelated to the suitability of the new method or methods being proposed. This is the concept of substitution. And stability indicating, for example, we pulled a definition from WHO TRS on vaccine stability, quality parameters that are either direct or indirect indicators of vaccine efficacy or safety, that are sensitive to storage conditions. And these parameters are used in stability studies and, of course, at lot release to control product quality throughout the shelf life. Determination of these parameters should result in quantitative values, and a detectable rate of change should be evident from those methods. So you'll see this terminology turning up throughout the talk. So updating stories. Here is one particular one, which I mentioned at the beginning, the histamine sensitization test. It's been around for a long time. It is to measure the presence of pertussis toxin, which if you do not have a complete toxoid process or an unstable process will turn up in the product. However, we'll discuss the problems with that, or the not such a problem with that. So the test was introduced in 1981 in Japan. It was in the European Pharmacopoeia as of 1991. The basis of the test is mice are naturally resistant to histamine, but with the exposure to Bordetella pertussis, they reduce or decrease their LD50 to histamine by up to 300-fold. And of course, that whether it's Bordetella pertussis or whether it's purified pertussis toxin, it has the same effect. So they're both lethal and temperature endpoint assays. Groups of mice are injected with different doses of pertussis toxin or test vaccine. After 4 to 5 days, animals are challenged with histamine, and the readout is either mortality or a temperature change. The limit of detection for the assay is 1 to 2 international units per dose. But there are at least 6 different versions in literature you'll see about how this test is to be performed in various jurisdictions. And if you're a manufacturer, that means potentially 6, if you're manufacturing in multiple markets, 2 or 3 or more different types of tests you must use. And each of these tests come with a liability. And as with all in vivo tests, the HIST is a highly variable assay that can result in false positive and inconclusive tests, which causes many repeats and delays of lot release, and in some cases even results in shortage of material. So that's on our balance we saw at the beginning. We're on the wrong side of that balance if we're using assays that can potentially result in product shortage. Vaccines are always in tight supply on the world market. Such a test really is not scientifically justified unless no alternative strategy is available. Yet this HIST test persists to this day due to the belief by certain regulators, at least, that the HIST is central to maintaining safety in pertussis. These are all competent and intelligent regulators, yet they believe something which is demonstrably not the case. And we all fall into these traps, by the way. So if considerations for the HIST continued, there were several methods developed for adjuvanted final bulk product tests, which is required by some regulatory jurisdictions. One of those final product tests was actually under consideration to be included in the European Pharmacopoeia. It had a similar limit of detection of 1 to 2 units. But a few things here. Pertussis toxin has no defined LD50 in humans. It's very different from the tetanus and diphtheria toxins, which are very nasty toxins. If you are exposed to chronic amounts of pertussis toxin, you go through a wasting condition, which can be reversed. And it was actually even used historically as a therapeutic in diabetes, but was found to be not effective over the long haul because it's so immunogenic, which is why it's not such a bad time to have in your vaccine. And in fact, wholesale pertussis vaccines had up to 100 to 300 units per mil, and that was not responsible for the reactogenicity of those products. The rat LD50 is 2,000 to 1,700 international units per kilogram body weight, and when we also, as it turns out, have a very sensitive test that's already in use in manufacturers, which has a limit of detection much lower than the HIST test. And that is in general use. So that assay is the Chinese hamster ovary assay, CHO cell, and it was described in 1983, so it's been around a very long time. And the idea is that when CHO cells are exposed to pertussis toxin, they cluster. This is a very visible difference with a microscope. This is a semi-quantitative test, but these cells don't die, which again speaks to the low toxicity or really non-toxicity of pertussis toxin. In the presence of pertussis toxin, they cluster. And the sensitivity of this assay, the limit of detection, is 0.006 international units per dose. As I say, it's a widely used test. And so the basic story update here is that while we do not need the HIST to control these products, it can be done at drug substance levels, provided those drug substances are demonstrated to be stable over their shelf life, and they will not revert. And if that is the case, this drug substance test is perfectly good. And this is a huge change in mindset around pertussis toxin and the assays required for that. So if we have this situation, there really isn't a final product test required. And as you'll see, that was deleted at the European Pharmacopoeia. If you want a more detailed presentation, my colleague from Health Canada, Richard Isbrucker, has a lovely presentation that he gave in London last fall. And the reference for that is here. You can go online and watch that, and you'll get a far better insight into that I've been able to share with you here. So updating the stories, again, new narratives. There was a meeting in 2015 at the EPA, organized by the EPAA in Europe. This workshop is published in Biologicals, and the reference for that is here. This meeting started because of the 2015 deletion of the general safety test by the FDA. That was seen as a great step forward. In Health Canada, we had not been using that test actually since the late 2000s, and that's true in some other jurisdictions as well. Industry, in this case, it was Sylvie Ulrich who made presentations showing that for the general safety test, there were, again, multiple versions of that test in different jurisdictions. These tests are slow and cumbersome, and again, were resulting in product shortages and had no real value to determining the safety of these products. The Paul Ehrlich Institute had a presentation which they'd shared at Group 15 a number of occasions as well. This was Klaus Kussler, now retired. Sylvie is also now retired from Sanofi. Klaus gave his very elegant presentation, which demonstrated that the scientific relevance of the general safety test had been demonstrated. The lack of scientific relevance was demonstrated since the 1930s, yet this assay had persisted. And Paul Stickings from the UK's NIBSC at the time, now the MHRA, provided some in vitro examples of ELISA detection of diphtheria and tetanus potencies, which were more sensitive and more reliable than the animal assays, and Paul will be speaking later today and presenting that data. So the key GST and diphtheria tetanus potency examples of the EPA were very compelling direct evidence to bring back to Group 15, and that helped drive the progress of 5214 and helped change minds within the larger Group 15 and Group 15V group working groups, and that helped deliver both 5214 and have it come out into the public domain, as well as this meeting was also an inspiration for VAC2VAC, which my colleagues will be speaking to later on in this meeting as well, a very important consortium. So what does 5214 actually state about in vivo and in vitro assays? Now, these are some direct quotes from the guidance, not simply my paraphrases around it, that all QC methods should ensure the comparability of the quality attributes between commercial batches and those batches originally found to be safe and efficacious in clinical studies or for veterinary vaccines and target species. However, the inherent variability in vivo assays make them less suitable, that's my emphasis here, than appropriately designed in vitro assays for monitoring consistency of production and for assessing the potential impact of manufacturing changes. As a result, it is essential to continually challenge the scientific value and relevance of these in vivo test methods. Now, these are direct quotes from 5214. So these are regulatory statements about in vivo assays. Very important note. The use of appropriate in vitro methods enhances the predictability and release of safe and effective vaccines for use. Again, direct quotes from EQM is not a regulatory group, of course, but it is a group which defines the guidances which regulators use both in Europe and around the world. So Group 15 moved past this fear of loss of animal assays. This was a result of an evidence-based discussion. Long-standing beliefs, which I would now call myths, were challenged and put aside. A really remarkable turnaround within the group because this was a very long and protracted discussion. So the key elements continued here. The primary focus of the implementation of any proposed in vitro method within a QC system would be the scientific relevance of the in vitro assays for the control of the critical quality attributes. And while in the European Pharmacopoeia, in vivo assay replacement within in vitro assays is typically achieved following multi-center collaborative studies, this should not be a prerequisite for individual products. So that could be happening then directly between a manufacturer and the relevant national authority. It needn't have a collaborative study, but it, of course, is greatly supported by collaborative studies, but it's not a requirement. While it may be desirable to have assays that have a wide applicability to a class of products, this again should not be a requirement. It is not the case for biopharmaceutics, for example. Manufacturers come in with assay-specific methodologies, and they are approved based on the suitability of the method and its fit for purpose. Those are the criteria, and we should be applying the same criteria in vaccines. In some cases, an existing in vitro method may need to be substituted by more than one in vitro method to characterize the quality attributes and the quantitative attributes measured by the existing tests. So not a problem. If it requires more tests, each of those tests, of course, need to be demonstrated to be fit for purpose and fit the overall strategy. So other considerations within 5214 for potency assays, the design of suitability stability indicating assays or combinations of alternative methods to capture CQA related to the potency is discussed. The fit for purpose principles, as I've mentioned, are discussed. For safety assays, considerations for different types of assay are presented for specific toxicity, molecular consistency by next generation sequencing, and Laurent will be giving a talk on that, and detection of viral extraneous agents by molecular methods, and that again is to the NGS topic. So what was remarkable about 5214 is the decades of stagnation. The 5214 really broke the dam within European pharmacopoeia and Group 15 and Group 15V. And one of the key and most delightful examples I can talk to, and Patrice Roux will be speaking about this example. And this is an example of while there were collaborative studies to support this, this is an assay that existed within Sanofi Pasteur for many years, and it is now has the first approval of a human in vitro assay for the control of rabies vaccine, which was just in the fall of last year. A huge breakthrough and a really welcome change. The abnormal toxicity or general safety test was removed from the European Pharmacopoeia. I'm not going to go through this complete list, but what you see here very clearly is the removal of a great many tests, at least in the safety area. We're still in the process of working on the potency side of that, and Paul will be speaking to that in a little bit. But we're coming down to these potency assays, and now we're into rabbit pyrogenicity, which Emmanuel Chancran will be speaking to today as well, and the use of the MAT. And I'll just stop here as well. There was a meeting just a few months ago in the U.S. where rabbit pyrogenicity and its removal and replacement by MAT or the bacterial and toxin test, the LAL test, were discussed. And there was very interesting speculation about the concern with the recombinant version of the BET assay is that we're actually endangering horseshoe crabs by the use of that assay as opposed to rabbits with their rabbit pyrogenicity. Of course, the recombinant's being encouraged, but as we develop our technologies and characterization, which is a hugely important idea to put on the table here, and was a big benefit of the VAC2VAC program, which will be described again later, is with the advancement of technology, we will perhaps be able to simply characterize by physical chemical methods pyrogens in a product, and we can even dispense with the MAT. And the MAT should only be used where the LAL is not applicable, by the way. It's another important consideration. I've listed here 3 adventitious agent testing documents, which are not a result of 5214, but were at least the first 2 of them were going along at the same time of 5214. So the non-animal approaches within these adventitious agent testing were greatly supported by the discussion that was going on around about 5214. And then we have mention of the VAC2VAC here for the diphtheria and tetanus potency assays. And I've also mentioned previously the COVID vaccines, which again were not part of 5214 at all. 5214 precedes the COVID vaccines by a long shot, but none of those vaccines were involved animals as in terms of the quality control of the products. And another wonderful, very recent development is that WHO, in addition to being inspired to remove the general safety test back in 2015 from A guidance and encourage that and ensure that wasn't perpetuated going forward, is now just convene a drafting group at WHO to develop a 5214-like document, a TRS for WHO. So this will go to a world message based on the success of 5214. So the main message here is that rigorous scientific debate and questioning of beliefs led to remarkable changes at Group 15 and Group 15B, and both groups now expeditiously amend monographs and general chapters to fit the science, which supports the innovation and change both in the Europe and now more broadly in the world. So next steps. At 5214, at European Pharmacopoeia Group 15, we'll continue to apply these principles to ongoing discussions around existing testing. With the increasing acceptance of 5214 principles by regulators, by WHO, manufacturers are more likely to develop in vitro methods for vaccine characterization, a very, very key step. Unless you have sophisticated characterization of your products, you won't have the tools to develop new assays. And now increasingly those groups those manufacturers can use substitution in addition to assay replacement and other approaches to bring those assays to authorization. Global acceptance of the appropriately developed stability indicating in vitro QC strategies is essential given world market. Hence, your participation in this event helps make that possible. Your understanding of these changes and the support for them is really critical to making that happen. So thank you for joining the meeting. And the use of non-animal vaccine characterization of QC is essential for that. It was essential for the pandemic, which of course was an enormously rapid and successful rollout of vaccines, all of which use non-animal quality control methods. So all the above support the use of appropriately qualified, I can't stress that enough, in vitro QC strategies for effective and robust in vitro control of CQAs for vaccines. So I thank you for your time, and I'm very open to questions.
Thank you, Dean. Let's wait a minute just to wait if questions are coming in. Thank you very much for this important overview that set really the scene for all the case studies that are coming in. So I just wish to remind people to put any questions in the Q&A, and we will be happy to either reply it today during this specific moment of the Q&A or in written.
So yes, the slides will be available after the meeting. We can respond to that.
Absolutely.
and as Laura mentioned, if for those of you who are feeling either all your questions were answered by the talk, or that you're a little reluctant to say that, as Laura mentioned, we're always open to pick these up after the fact and respond. And please reach out. Our contact information is available through Laura, and we'll be happy to address that. Great. And I will stress, sorry, I'll just repeat one thing I've mentioned, is that while the ethics considerations around animal use are essential, even in the preclinical space, of course, if we don't have humane considerations for animals, the data we get from those animals is a waste, in addition to it being just massively inappropriate. However, those considerations are different in different jurisdictions, and whereas the science approach to this is what has made the difference for 5214, and that is a universal dialogue regardless of the considerations in any particular jurisdiction. So we find this a more universal message, and hence the success of 5214. So I'll stop there.
Dina, we have a request through the chat saying that there is the recognition that the WHO is still a little bit weak in terms of the implementing of the triage concept in general, and there is a question about where are those gaps? So why there are still those gaps? If you can.
Sure. So I did sort of touch on this. There are gaps, not because we don't have the technologies, but the gaps are there because industry is reluctant to invest in those technologies if they don't see a path forward. So I mentioned, I'll go back to the rabies example again. If you're an industry that knows there are regulators across the table who are smart and capable people, but have this belief that only the rabies NIH test is the safe way to develop that. And again, this is a life and death vaccine. So if you're a clinical evaluator or a quality evaluator institute, and you sincerely believe that the only reliable way to get that product out of the door that's going to be safe and effective is an animal test, then you're very reluctant to change that test. But it is demonstratively wrong, and Patrice will speak to that you can have, and we have had, multiple methods of doing that, and Sanofi settled on a particular one and had that approved, that actually give you better quality control in the rabies space with their assay, much more sensitive quality control and of various quality attributes of a product. And Paul Stickings will speak to that with his D&T test. So while there may be some gaps in certain areas, they are merely technical and resource. If industry understands that regulators are open to these changes and will look at the data and not be driven by their fears about this, will simply evaluate the methods as fit for purpose, the scientific rationale, they will quickly dismiss it. And again, the HIST is another key one. We could have removed the HIST test decades ago based on our understanding now of the very low toxicity of that. In fact, it's even potentially useful to have in a vaccine. That's not true of other toxins, but it is true of pertussis. And the control strategy, which we are now using, by the way, Canada has implemented some years ago. We no longer require the HIS test, and we did not require the reversibility test since the early 2000s, and that has been in place. And I will tell you that, but I know at least one regulator, and I know there are multiple, who at the time we started this work absolutely believed, not true, but absolutely believed that if we didn't have the HIS test, babies would die. That would be a quote from the lips of some of these people, and it's simply not true. So what 5214 is about changing the hearts and minds of people, and it's up to regulators to be aware of the reality of data as opposed to the perceptions of reality through these myths. That's the success of 5214. But the excellent question, and thank you for that.
Thank you, Dina. We have actually an interesting one as well that you already kind of replied. That's seeing that all different stakeholders that are involved into this transition, into this major transformation. In your view, who is the most resistant to change?
Who is the most resistant? Well, you know, I've learned something very recently, which surprised me in discussions around COVID vaccines and assay control strategies, that it came to my attention that even certain individuals within certain companies, and of course, I'm not going to name them, they'll themselves also have the idea that animal assays are very appropriate for control of vaccines. So it's not unique to regulators. And this is an important realization that we, you know, there is again another sociological piece here. As I said, this is more of a philosophical talk. People should be aware of the Dunning-Kruger hypothesis, which is the more we know about something, the less the less we relate ourselves expertise, the less we know about something, the more we evaluate our expertise. So if we haven't dug deep into these thoughts and we rely on hearsay, we think we know what we're doing, but we actually don't know at all. The deeper you get in, the less certain you are about the reality situations, and that's a comfortable place to be. Be more open to new thoughts, and this applies to every single one of us. It's not about intelligence, it's about perception. So again, if you want to see this thing drive forward, the blockade is people's perception. The reality, as you'll see from Paul and Patrice and others, is very different from those perceptions, and that's up to all of us, industry and regulators alike, to be deep on those realities, and always challenge our own mindsets. That's, I guess, the take-home message.
Thank you, Dean. And in the interest of time, I would move to the next presentation. We'll try to answer to the remaining questions through the next case studies. Thank you, Dean, for being the opening of this webinar. And it's my pleasure to introduce you, Dr. Joris van de Putte and Dr. Carmen Ljungbeck, that they are going to present about the back-to-back project, which is actually the one that also Dean mentioned, that it is a very important example of multi-stakeholder collaboration. So, Joris, Carmen, the floor is yours.
Thank you very much, Laura, and thank you to the organizers for considering a very brief overview of the VAC2VAC project that our friend Dean mentioned at least already 10 times in his presentation. Thank you very much, Dean. Dean, you were part of the advisory board together with IDUKM and Robin Lewis, who is also present here. So the VAC, can I have the next slide, please? The VAC2VAC project started March 22nd, 2016, a date I will personally never forget. And VAC2VAC is very importantly an innovative medicines initiative project from the European Union. The IMI, as we call it, is always composed of a participation of EFPIA, so the industry, the pharmaceutical industry on the one hand, and then European Commission, the European Commission that then funds the official institutes, the science, and other organizations. So it is very important that the philosophy of IMI is really a real collaboration between industrial partners and official partners, regulatory partners, and official laboratories. Can I have the next slide? Why telling that? I must say that all those who worked in Vac2Vac from 2016 to 2022 are still very excited about this huge collaborative effect and this huge collaboration. It was like a team between industry and the partners, and dialogue was very important, and we could say openly the things that we liked, we did not like, and then conclusions were taken in order to progress. And as you will see, I think we can all be very proud about what this Vac2Vac consortium realized. So, sorry, 21 partners, 15 public partners, 3 FPIA partners, and 3 also animal for health, so companies who are active in vaccines for animals. This is a total budget of about 16 million euros, of which the EFPI partners contribute in kind. The next slide, please. So the Vac2Vac partners themselves, you see here the list. So it's in detail what I told before. Very importantly is that we were scattered all over Europe, and we have all the different interests and all the different competencies and experiences have been present from the industry towards the national reference laboratories like NIBC, RVIM, AGES from Austria, the Italians, ISS, PI from Germany, and Sensano from Belgium. So, and then we had some vaccine alliances, EVI and IBSAU. By the way, I like to thank EVI. The first coordinator of EVI was Odile Leroy, and then followed by Hilde de Prader, who both did a tremendous work in keeping this consortium very well together in a very smooth way. So then we have a regulatory agency, the Dutch MEB, then the European Reference Lab, the GRC, and then academia and transnational organizations like BPRC, Intravac, and there you see the representatives of the different countries. The next slide, please. Importantly is that I think it's after the ZAPI project, one of the first projects in IMI or even in European projects where there is a real One Health approach, starting from the philosophy that animal health industry can learn from human health industry and human health industry can learn from animal health industry. And we had a lot of interesting exchanges between the two. Next slide, please. So, very importantly, before we started was to recognize the reality of in vivo testing. Dean told already a lot about that, so I will go quickly. When we started, we estimated that worldwide there were more or less 10 million animals that were used per year just for batch release control. And then if we know that, that these tests are quite variable to highly variable, these are really elastic tests where it's difficult to control against shifts and also results that can drift or shift from just the simple supply of breeds of animals. So the predictability for potency is really questionable, if not predictable. They are time consuming, they are costly, and because of these reasons, it blocks or hampers vaccine availability when we know where the vaccine availability need in the world is so high. So as we will see, The in vitro alternatives, like Dean already insisted on, are consistent, reliable, reduce time and cost, and increase the availability of vaccines. Can I have the next slide, please? So I will give in the 2 slides that come now. First of all, examples of what has been realized in the VAC2VAC consortium on the in vitro potency. So comparing in vitro to in vivo, there is a very interesting study that has been published by Stalpers and others about the variability of in vitro potency tests for DTaP vaccines. And there we have seen a variability in that study that ranges for 16% to 132%. What means, well, the vaccine batch that is excellent today can be declassified tomorrow. So then what VAC2VAC did is, in order to try to come to in vitro quality attributes that are well characterized, that are more reliable. And as you will see, Paul Stickings will go into detail about the enormous results obtained in DTaP vaccines, with in vitro variability of less than 10%. For the tick-borne encephalitis virus vaccines, an ELISA has been developed by the different partners. So when I talk about the partners, it's a real collaboration again between industry and the other parts, so the research, the science, and the laboratories. In veterinary rabies, there has been confirmation of the high consistency of the ELISA glycoprotein detection. And further, there have been interesting results on the clostridial vaccines, particularly of Clostridium shovi, which showed a high specificity to differentiate degraded from non-degraded vaccines. The next slide on safety, please. So Dean told a lot about the abnormal toxicity tests and the safety tests, which were all done on animals, how inefficient and they are, and how the need is high in order to replace them. So it's interesting to say that our German colleagues, one of the German colleagues of PI, made a very good historical overview between the initial reasons of those kind of tests in the beginning of the 20th century, so 1910-20, and then the continuing, because this belief about which Dean told, this belief, well, we are happy with the test, and the test was then used for, in fact, proving things where it had no relation anymore. So in VAC2VAC, the achievement is that for Clostridium perfringens vaccines, to prove the absence of residual toxin was done on THP1 cells. Clostridium tetani in human and veterinary vaccines went also to in vitro testing. Dean talked already quite a lot about the MAT test to replace the rabin pyrogen test. And then in the field of animal health, feline leukemia vaccine, a very good example is the test safety on THP1 cells. Next slide, please. So VAC2VAC had also a quite important part on regulatory. And with our regulatory team, we went out during the consortium time, and we are still continuing together with Laura, together with Health for Animals, together with EFPIA, to bring the results globally. So what we have seen, and I repeat just here what Dean said, is the fear factor. And the fear factor, it's incredible. You see it with regulators in industry and also from political side. Because the reason try not to change is hugely variable. Therefore, in our actions now, we propose a stepwise approach. So meaning that there can be local reasons why people refrain to come to in vitro testing. And the first thing we have to do is to really listen, and listen again. Why? Then the answer is, of course, only scientific-based data, and then you go into dialogue. That's what we try to start now with post-fact-to-fact activities. So, and there are already examples. The 3 examples named by Dean is, first of all, the COVID vaccines have all been batch release tested by in vitro testing, the HPV vaccines and the conjugated meningococcal and pneumococcal vaccines. Can I have the next slide, please? So this is the last slide that is coming now. As a conclusion, what we have seen in the VAC2VAC, and it's not only VAC2VAC, but this consortium was really important in order to stress the importance of current GMP production methods and the consistency approach. So we have seen that in vitro methodology is really compatible with in-process control and incompatible with the consistency approach. By this approach, non-conforming batches can be detected even much earlier before the date of batch release. And as we have said, but I repeat it, the in vitro testing avoids great elasticity and variability of the in vitro testing. And when it comes to replacing in vivo by an in vitro test, a one-by-one is not always possible. It's even not needed. It's rather better to look at historical results because by definition, since the in vivo testing is so variable, you cannot justify really the one-to-one comparison, particularly post hoc. So it's important to think globally, and I will not continue to dwell on that because it's important for all parts in the world which want now to become global partners in the vaccine access. So it's in the interest of all partners, where they might be on this globe, to come to a harmonization on the same methodology for batch release and same methodology for in vitro testing. An important lesson that we learned, probably the most important one, is the one of dialogue between all stakeholders. I stop here. Thank you very much, and I am available for your questions.
Thank you, Joris. Let's see if we have some question. Okay. Thank you, Mustafa, about the whole cell pertussis. We will get there. We will get there. Let's see if it's in the chat we have some additional Q&A. Okay, I guess, Joris, that we can move ahead to the next speaker. Thank you very much for your
overview about
the VAC2VAC project that has been really a very important example of collaboration, also from method development, but also from the regulatory transformation. So thank you very much for presenting the project. And our next speaker, it has been already called out more than once. Thank you, Paul Stickins from HRA UK. And Paul is actually getting into our first case study for this webinar, digging into what the VAC2VAC project has achieved with regards to the non-animal methods for the potency test of diphtheria and tetanus vaccines. Paul, thank you for being with us. The floor is yours.
Thanks, Laura. Can I just check you can hear me okay?
Yes, yes.
Perfect.
Thank you, Paul.
Okay, good, thanks. I am, however, on a hotel Wi-Fi system that I don't fully trust, so I am going to turn off my camera just for the purposes of the presentation, I will put it back on for the questions. So good morning, afternoon, evening. I have the pleasure of talking you through in a bit more detail one of the outcomes from the VAC2VAC project, and it was related to the development of an immunoassay for potentially substituting potency tests for the diphtheria and tetanus vaccines. My slide, there we are. So potency testing is a regulatory requirement for the release of every lot. Potency is a critical quality attribute that is a test of the functional integrity of the antigen, and it provides important assurance that the vaccine lot will elicit the desired immunological response in the target species. It's usually a very important indicator of stability of the vaccine as well, and it provides important assurance for consistency of the manufacturing process, so that evidence that new lots that are produced are comparable to those originally used in clinical studies or through routine clinical use. And there are different approaches that are used for potency testing. Starting on the left, we have challenge tests, which require challenging an immunized animal with either the pathogen or toxin that is relevant to the vaccine in question. And the best examples are the diphtheria, tetanus, and pertussis potency assays. Rabies, the historic rabies NIH test is another example of that. And there are many, many cases where these challenge tests are still in use. There are also serological models which still require animals. They are immunized but not challenged. Instead, they're bled and the antibody responses in the blood are measured. And those models exist for diphtheria and tetanus and acellular pertussis, for example. And then we have examples where animal tests were introduced and used for routine potency testing at the time the vaccine was licensed and first introduced, but subsequently was then replaced with an in vitro assay. And hepatitis B for human vaccines and Newcastle disease vaccine in the veterinary world are examples of that approach. And lastly, we have vaccines for which an in vivo assay was never used for the routine potency testing, and instead in vitro assays were implemented at the time of licensure. They were developed throughout the early R&D and the non-clinical development process, and then used routinely for potency testing. And the best examples of that would be things like the COVID mRNA vaccines, for example. And thinking back to DTP, there is a lot of animals needed to fulfill that routine potency testing function, because every lot must undergo the test. Of course, it must pass the test before it can be released. And animal tests, whether challenge or serological, are used for each of those components, D, T, and P. There isn't anything in the public domain that I could find about the number of animals globally that are used for this activity, so for routine potency testing. And I think that's because it's not an easy thing to calculate. We can make a very crude estimate, which is what I've attempted to do here. And it's based on some information that is in the public domain. So number of live births, for example, the global coverage with DTP vaccine, and that means 3 doses of a primary series. So we can estimate how many doses are needed, but then we have to start making assumptions. So for example, the batch size, I've assumed a figure here for a batch size, but it will definitely be smaller for some producers and it may be larger for others. So it's an assumption, and that will give us an assumptive estimate of the total number of batches that are needed. And again, more assumptions about the number of animals in each assay for each of those components. But if we put that together, we would get a figure of about 140,000 animals needed routinely per year for the post-licensure potency testing. Now, I think that's probably a conservative estimate because I haven't accounted for a number of things, retests, independent testing done by some control authorities. I haven't counted for veterinary tetanus vaccine assays, for example. So I think it's going to be far higher than that number shown, but I show it just to provide a bit of context and to give an idea of what we're hoping to impact with the development and introduction of assays that could substitute for the current potency tests. And, there are very good reasons to try and do that. And we've heard already some of these. The ethical concerns, clearly very important. There are resource intensive and economic costs that are associated with animal testing, and they take quite a long time to perform. So from the start of the test to the time that you get the result can be at least 6 weeks and more if you have to do a repeat because of an invalid test. And that can potentially impact supply chains for vaccines, which is a very important consideration. And the last point here on the list is starting to receive more attention, as it should. And that's the fact that the in vivo assays are not the best model for routine control of vaccines because they are highly variable and they have a poor discriminative power. So if we're interested in understanding consistency of production and we want the ability to identify quality differences between batches, they are not the best tests to be using. And there's a really nice paper that's been published that quantifies just how variable some of these assays are, and I show that here. And then at the bottom, I'm showing some data from Emmanuel Coppens from Sanofi, who presented this at a previous 3Rs meeting some time ago, but it's such a striking example that I keep using it. And it shows that for a tetanus vaccine on the left and a diphtheria vaccine on the right, if you deliberately reduce the amount of antigen in the vaccine, you have to make quite a big change before that vaccine no longer conforms with the current requirements of the potency test, which is really striking, and it tells you something about how discriminative these assays are not. So what we've done in the Vac2Vac project is develop a monoclonal antibody ELISA that is intended to provide a relative quantitative estimate of antigen content using antibodies that we know are directed against relevant epitopes and that are sensitive to changes in the quality and the integrity of the antigen. So it's a very typical sandwich ELISA format that I think many people will be familiar with. And if we do a very quick side-by-side comparison of the key characteristics of a typical animal potency test and the monoclonal antibody ELISA, we're looking at a test that is much faster, doesn't require animals for the performance of the test, has much better precision of each estimate, and therefore is much better in terms of discriminating between batches. And just a word again about how such an assay would be positioned in a product life cycle. Starting at the top, I'm reiterating what has already been said by Dean, that in vivo assays do play an important role in early vaccine R&D and in the early non-clinical development. And for newer vaccines, in vitro assays should, and I think are, developed alongside these animal tests such that they can be taken forward for routine use in the control strategy post-licensure. But that isn't possible for legacy vaccines such as DTP, because at the time those vaccines were developed, we didn't have the tools that are available to us now. And I think at that time, it was the right thing to do to take those in vivo models through to the post-licensure control strategy. Now we do have better tools, and therefore what we are looking to do with the type of assay that I'm going to show you in a minute is to substitute at this stage the routine QC lot release stage for those legacy potency tests. Now in the project we had, you've already seen, we had a number of manufacturers. Those manufacturers have a number of vaccines, and we looked at a lot. So we looked at a really wide range of products from the human side, from the veterinary side, different formulations, different combinations, different adjuvants, and I cannot show all of that data. So I'm going to show representative examples, but what I show you, we consistently saw the same type of result across those different product types coming from different producers. And you get a sense of that just with this first figure, which is the diphtheria monoclonal antibody ELISA applied to a range of different diphtheria containing vaccines. There's also some drug substances in here, pre-adsorbed, which is the black line, and adsorbed, which is the red line. And then all of the other curves are for drug product, so different combination vaccines, all tested in the drug product as presented, so no modification. That means tested in the presence of the adjuvant. Then we did some studies to look at sensitivity and specificity. So in terms of sensitivity, we were interested to see how small changes could or could not be detected by the assay. So I'm using an example from the diphtheria mAb ELISA, and we have got vaccines deliberately formulated to have 25% reductions in the antigen content. And you can see quite nicely that the assay is able to distinguish between those different samples. And if you think back to the slide with the data from Emmanuel that I showed, where you had to make very dramatic changes in the amount of antigen before the animal test would fail. So we're dealing with a sensitivity here that just doesn't exist with the animal models. And then for specificity, the black line at the bottom is the vaccine with all of the components and the excipients and the adjuvant, but without the diphtheria component. And you can see we have no signal whatsoever. So we have very good specificity as well. We extended the range of antigen content by spiking antigen into a vaccine, so up to 400% of the target concentration, just to ensure ourselves that we had good linearity of measurement. And you can see quite clearly that we do. And this is an example from the tetanus mAb ELISA. We saw exactly the same thing with the diphtheria assay. Very important to look at what the assay can detect in terms of quality and integrity changes. And the most common way to do that is to deliberately stress the vaccine by exposure to heat. And that was done for a huge range of different products. I'm showing an example of a veterinary tetanus vaccine this time with the tetanus ELISA. And you can see the blue is the normal storage conditions for that product, and a clear shift in the assay response as the temperature is increased. So that's a really nice example of how the assay can identify changes in quality that are induced by temperature stress. Gives us some evidence that it's likely to be stability indicating. And the other thing we wanted to understand, because we're dealing with an assay that is going to be sensitive, we wanted to see how it would work with real world samples where you're dealing with drug products coming off the production line that can have antigens of different age, because the shelf life of the upstream components can be quite long. And when you put that all together into the drug product, the age of the toxoid can actually vary quite dramatically. And we wanted to understand whether the ELISA would be impacted by that. So what you can see, the x-axis is the age of the toxoid in the vaccine in months. So this is a long period of time that we're looking at. And we've got batches coming from all of those different time points with a reference batch somewhere in the middle. And you can see that there's no evidence of an age-dependent impact on estimates of relative antigenicity, which gives us some good assurance that this assay is likely to be fit for purpose in the real world. Then we wanted to assure ourselves that the assay is robust enough to be transferred to other laboratories. And we did this with both the tetanus assay, which is what I'm showing you here, and the diphtheria assay. Here we had 5 different labs from both the human industry and the vet industry, covering lots of different product types, different formulations, different adjuvants. And we set the target for the intermediate precision to be 15%. And you can see that in each case, both the receiving lab, so that was the industry partner, and the donating lab, that was my lab, were able to achieve that level of intermediate precision. And in fact, in most cases, it was below 10%, and in many cases below 5%, which is really, really good for this type of assay. So the work that I've presented for the diphtheria assay is now published. The tetanus side of things is under peer review. The manuscript has been submitted, and we hope that that will be accepted and in the public domain very soon. I should also point you towards some very important work that was done by another partner on the VAC2VAC project. This is the Control and Regulatory Authority in Belgium at Science Sarno, and they have used a similar approach to what I've described. So it's monoclonal antibodies, but importantly, they have multiplexed the assay using Luminex technology such that it covers not only D&T, but also the acellular pertussis antigens, which clearly is very attractive for some producers who will want to follow that strategy and who have those combinations of vaccines. What we did in the project is not fully validate the method. We've shown proof of concept. We're trying to provide the evidence that this is a strategy worth pursuing, and I think we did achieve that. And it's now really up to manufacturers, and certainly the ones that were part of that consortium, to further explore the utility of these approaches in the context of the products that they are making. The second to last very important thing I want to share is that the antibodies, the monoclonals that were used in the work that I've shown you and are also used in the work that Science Sarno have done, are all available through the NIBSC catalogue. And that is testament to there are 4 different organizations who own those antibodies, and they all collaboratively agreed that they should be made available to people who want to develop and implement these assays. And that removes one of the most common barriers to furthering 3R's approaches. And thanks again to those organizations involved. And just the last thing I want to mention briefly, and I saw the question in the Q&A about whole cell pertussis. Now, the VAC2VAC project was a European initiative, and it focused on vaccines licensed in Europe, which are predominantly acellular pertussis combinations. So we didn't look at whole cell vaccines within the consortium, within the project. However, most of the world's infants do receive a vaccine with a whole cell pertussis component. And so most of the animal use for DTP potency testing is related to whole cell containing vaccines. What we've done since the project is we have had a look at how the D and the T assay performs with a whole cell vaccine. So we've only looked at one product just to get an idea. I've shown the example here on the slide, and it looks good. It looks as if the whole cell component itself isn't going to interfere with how the the D or the T assays perform. I mean, we need to look at more products, of course, to fully understand that. But looking at the whole cell component itself is clearly going to be more challenging, and there is definitely more work to do there. And I would encourage people who can help make that happen to work together to really maximize the impact of this approach. Because if we don't cover whole cell pertussis, we're not going to achieve the impact. I'd like to thank all of my colleagues and other partners on the project, and the industry partners, of course, who were instrumental to getting all of this work done. And I'll happily take questions if we have time, and I'll address any in the chat that we don't have time. Thank you.
Thank you, Paul. Yes, we have some questions around. So I guess that's one of the most important ones that I would choose for this specific moment is about the possibility to collaboration to transfer what has been done by Back2Back to other companies or organizations. Again, in view of prioritizing the introduction of non-animal testing for DTP products. So if there are any plans or any ways that organizations, public, private, that were not part of Back2Back can collaborate or transfer, or maybe consider the transfer of the methodology that you have developed?
Yes, so certainly speaking for the mobilizers that we developed in my lab, we're very happy to support the transfer of those to any partner, public or private. As I said at the end, the antibodies are available, so that's a key aspect. One of the methods is published, the other one should be published very soon. So everything is there for anybody to make a start setting it up. And we are very happy to provide the SOPs that we follow and to have technical calls or provide the support that anyone needs if they go through that process of trying to set it up. I obviously can't speak for the multiplex assay from Sciensano, but I'm sure that they would be keen to help people as well. As for any more formal initiatives in terms of wider studies or initiatives that are more formal, there is nothing yet that is in development. Whether there will be or not, I think it's a bit too early to say.
Mm-hmm. Oh, thank you, Paul. I think that is it's a little bit what we would like to achieve through this type of webinar and future conversation, to understand exactly what is the interest for all these stakeholders to perhaps collaborate together, and then we can think how to set up something that is more structured if we really see that there is a joint need. There are actually a lot of questions within the chat and the Q&A. Some of them are rather technical, so if it's okay with you, Paul, to just answer in written, so we can continue in the interest of time to not really go too much further into the agenda. But thank you very much for the work that you presented. It has been really nice to see what are the opportunities, the concrete opportunities for DTP. And thank you also for sharing the information on the whole cell pertussis that we definitely know that is unmet this need, if you can say, in terms of transitioning to novel technologies, hopefully in vitro. So thank you, Paul.
Thank you.
And again, thank you for the patience of our participants. We will try to address all the questions via the chat or the Q&A. Please keep posting them. They are very important, and we hope to answer to all of them. without further ado, I would like to introduce the next speakers and the topic. We are actually moving to another important example, which is the example of the human rabies vaccines potency test. And the speakers are Patricio from Sanofi and Enrico Teao from ADQM. Give me 1 second that I'm going to bring up your presentation.
Thank you, Laura.
Okay, let me know if you see it.
Yeah.
Okay.
Can you put it full screen?
It is. Let me know if you see it in full screen mode.
Can you put in presentation mode, please, Laura?
Is it now?
Yeah, it is. It is. Perfect.
The floor
Thank
is
you,
yours, sir. Thank you.
Yeah, thank you. Thank you. Hello, everybody. So today, myself and Enrico will present you the human rabies in vivo assay, in vivo potency assay replacement by in vitro potency. So I will start in the first part, and Enrico will follow in the first part with the Sanofi work, and in the second part, Enrico will discuss more the regulatory and international collaborative study part of the project and case study. Can you move on the next slide, please? So just I will give you a context, and also I will mention ELISA validation data. I'm sorry, but I will go very rapidly on that. I mentioned in a slide all the publication we have done on this ELISA development and validation. So you will be able to look at the data if you want more later in the presentation. But in the sake of time, I will go very fast. And then I will take a bit more time on how we define the potency acceptance criteria when switching from in vivo to in vitro. In 2 cases, a new rabies vaccine we are developing, and second for a commercial vaccine, which is currently on the market. Next slide, please. So in terms of context, we started our work with, we have in our vaccine pipeline 2 commercial vaccines that were registered more than 40, 50 years ago. And then we started our development of an in vitro potency assay for rabies in the context of the clinical development of a new rabies vaccine, which is called DRVG, and what is meant to replace both commercial vaccines that are mentioned on this slide. Can you go to the next slide, please? So as mentioned, the current in vitro potency assay that is mentioned in a regulation, which is compounded for rabies, is the NIH in vivo potency assay. I will not go into in detail on that also, but as mentioned by Dean before, this assay has been used for more than 50 years to release rabies vaccines. But the main drawback with this in vivo potency assay, which is that it is highly variable, and it uses a large number of animal per test. And in the context of the development of our new rabies vaccine, we started our work in order to replace this in vivo assay by an in vitro assay. Of course, we are also guided by the European Pharmacopoeia and European directive to support the replacement of this animal model. And as mentioned by Dean, later on, the chapter of 5 to 14 for replacement of in vivo assay was also a strong guide for us in our replacement strategy. And in addition to that, I just want to mention that within Sanofi, we have also a 3R policy that helps us replacing in vivo assay. Next slide. So this slide just describes the data that were available prior to our development work that was suggesting that G protein ELISA assay could be a good surrogate of potency. The major elements are that the correlative protection against rabies disease is the presence in the human sera a neutralizing antibody against the rabies G protein. 83% of this neutralizing G protein against the G protein are targeting a specific epitope, which is a domain 3. And importantly, the good 3-dimensional structure of the rabies G protein is key for developing and inducing this neutralizing response. And finally, prior to our work, we had several papers publication that were suggesting that there is a good agreement between the NIH test and an ELISA against the G protein that was pre-existing. So this was the work that was available when we started our development. Next slide, please. Okay, so our ELISA is based on 2 different monoclonal antibodies. a capture monoclonal antibodies, which is the 1112 antibody that targets the domain 2 of the G protein, which is discontinuous and that is conformational and associated with 2 disulfide bond. We use also the D125 antibody, which is targeting the antigenic site 3 of the G protein, which is also conformational. Next slide, please. We checked that these 2 monoclonal antibodies are indeed functional. So we demonstrated that the 2 monoclonal antibodies, the D125 and the 1112, were able to neutralize rabies vaccines, and we did it on 3 different strains, and using the refit assay. And indeed, both monoclonal antibodies are neutralizing antibodies. They prevent rabies vaccine infection, and thus they are functional. Next slide, please. So this is how our ELISA is based. So we are using the monoclonal antibodies 1112 as a capture antibody on a plate, on ELISA plate. Then the D125 antibody is a detection antibody. This detection antibody is coupled to biotin, and we are using streptavidin peroxidase for colorimetric assay in addition to OPD. So the ELISA, as mentioned, is using 2 neutralizing antibody targeting 2 different epitopes. And we are using for the assay the international reference or an internal reference calibrated against the international reference. And we started the implementation of this ELISA for our new vaccines, next generation vaccines, VRVG, and also we started to implement it on our commercial vaccine, which is called VeroRab. Next slide. The first data we started to generate is data to demonstrate that the rabies G protein ELISA is stability indicating. So we created experimental condition to demonstrate that this ELISA is able to demonstrate the degradation of the product and the vaccine when it is treated by heat degradation, inactivated by reduction alkylation, and also when the product or the virus is hyperinactivated by beta-propiolactone. Beta-propiolactone is the chemical component we are using in our vaccine manufacturing to inactivate rabies vaccines. And we know that when concentration of BPL is increased, BPL can degrade the antigen, and that's what we showed using our ELISA. So meaning that we generate the data to demonstrate that the Sanofi Pasteur rabies G protein ELISA detect alteration of the G protein, whether it's by heat or by chemical, and it is a stability indicating assay. Next slide. Then we demonstrated or tried to demonstrate that this rabies G protein ELISA is in agreement with the in vivo NIH potency assay, but more discriminative, meaning that we created subpotent lots either by thermodegradation, and we compared the ELISA and the NIH test results. And we also did subpotent lots by subformulation, and also by subpotent loss by hyperinactivation. And in each case, what we could demonstrate is that due to the much lower variability of the ELISA, the in vitro assay, it's much more discriminative. And I will take just one example on the slide. When for VeroRab, we created an artificially degraded product by mixing 50% of intact and 50% of degraded vaccines. The ELISA was able to discriminate between the inactivated and intact vaccine. For the NIH test, you can see that the value for the intact vaccine and the 50% in inactivated vaccine are not significantly different. So all these data suggested that this ELISA was a good candidate to replace NIH, and indeed much more discriminative. Next slide. Then we ICH validated our ELISA in our 2 products in different production steps, meaning drug substance and drug product, and we were able to demonstrate that the assay is specific, linear, accurate, and precise. And in terms of precision, we are around 10% or below. as compared to what we could know about the NIH test, where the variability, as mentioned by Dean Smith, is that between 25% to 400%, meaning plus minus multiplied or divided by 4, as compared to ELISA, which is more multiplied or divided by 1.1 or lower. So the ELISA is much less variable as compared to NIH. Yes, you can go on the next slide. So this slide just summarizes all the elements that suggest that this rabies ELISA is indeed a good candidate to replace the NIH potency test. It uses 2 different neutralizing antibody, functional neutralizing antibody. It is the ELISA is fully validated. The ELISA is stability indicating. The ELISA is in agreement with NIH, meaning it follows the same trend, but our data all demonstrate that the ELISA is much more discriminative as compared to the NIH test. Next slide. The last data we generated and we've published is this one. Because for our new vaccine, we were in clinical development, we've published the clinical data of a dose ranging study where we could demonstrate a dose-dependent relationship between the content in rabies G protein content by ELISA and the human immune response in neutralizing titer. So you can see that when we inject in human, increasing its dose of G protein as measured by ELISA, we could see a trend and a dose-dependent relationship with a neutralizing titer in human. When we use exactly the same lots and we plot the GMT, the neutralizing titer, towards the NIH test result, you see very low or not at all any relationship between the neutralizing titer and the NIH test. So meaning that the conclusion of that is that the ELISA can predict much more than NIH the immune response in human. Next slide. So I went very fast, as I said, but now we'll concentrate on the change of acceptance criteria. So for our new rabies vaccine, VRVG, we use the ELISA during process development. The G protein ELISA was implemented on drug substance intermediates to monitor yields and losses, and also to ensure the consistent quality of our antigen and of the G protein along DS process. The rabies G protein ELISA was used to formulate our vaccines in clinical trials. The rabies G protein ELISA was also used to monitor drug substance, both drug substance and drug product stability. And the VRVG ELISA was used to determine our clinical dose since phase 1 and all along the clinical development. And during our clinical development, in each product we injected in human, we also performed in parallel the NIH test in addition to the ELISA on the drug product. Next slide. Please. Okay. So this slide just summarizes the strategy we will be using to define the in vitro potency acceptance criteria for our future new rabies vaccine. For the submission of our CTD, the criteria will be based on clinical data, meaning they will be patient-centric. So the lower limit for potency will be based on the minimal ELISA dose demonstrated to be efficacious during clinical trial, and the ELISA upper limit on our drug product will be based on the maximal ELISA dose injected in human demonstrated to be safe. So this will be the ELISA acceptance criteria, and at release and at start and at submission. And during lifecycle management, we'll keep the same release limits, but we'll define internal limits much based on process consistency. So that's the approach. So basically, in conclusion, for our new vaccines, our release specification or acceptance criteria for potency will be based on clinical data and will be patient-centric. So this will be the strategy for VRVG. As I mentioned, the CTD is not submitted yet, so I cannot provide you with data because they are under finalization. So this is a strategy for a new vaccine. You will see in the next slide that for VeroRab, which is our commercial vaccine. Can you go to the next slide, please, Laura? Okay. For our VeroRab vaccine, which is currently on the market, the strategy to define the in vitro potency acceptance criteria is very different. It's very different because VeroRab has been commercialized for more than 40 years, meaning the clinical data we develop for VeroRab are very far away. So we cannot base our limits based on clinical data. But we use a consistency approach. So the consistency approach is based on the calculation of a new acceptance criteria with a mean plus or minus 3 standard deviation. And this statistical approach was based on representative batch of VeroRab, conform representative batch of VeroRab, and it was done on more than 279 batches, data generated on 279 batches, covering 3 years of manufacturing. So you see the data on the plot. So we were able to demonstrate the lower limit of 1.5 EU per dose, ELISA EU per dose, and the upper limit of 4.3 EU ELISA per dose. And these limits were confirmed in agreement study by generating subpotent batches that were tested both by NIH and by ELISA. So a strategy very different as compared to VRVG. Can you go on the next slide, please? Next, okay. So we switch and we define acceptance criteria for the G protein ELISA potency on DP. But in addition to switching from NIH to ELISA on a drug product, we also implemented on VeroRab the ELISA on intermediates, meaning we developed a new ELISA formulation target for our product. We used in the past for VeroRab another in vitro assay, which was called SRID. So we replaced SRID by ELISA. So we define a new formulation target for our vaccine. We also implemented ELISA on a drug substance in replacement of SRID to monitor stability, to monitor antigen content on DS, but also to monitor stability. And we also implemented the rabies G protein ELISA on drug substance intermediates. The advantage of the ELISA, it has a wider linearity range. It's less sensitive to matrix interference. It's more reproducible as compared to SRID. So the ELISA in the drug substance intermediate was more appropriate to follow process consistency and drug substance quality. And of course, monitor yield and losses. And the good news is that when we submitted our variation in 22 for VeroRab for the replacement of the NIH test by ELISA, the EMA gave us a positive answer, which was a great achievement for us. And currently for this product, our submission are ongoing worldwide. Next slide, please. And I think the last slide, if I remember well. Next slide. Okay. So in conclusion, what I mean is that for the replacement of in vivo assay by in vitro ELISA method and setting specifications, the in vitro suitability and validation data package is key. As mentioned by Dean, this data package was developed in order to demonstrate the suitability and the performance of the in vitro assay as compared to the in vivo assay. We also considered monitoring, replacing the in vitro assay on DP, but not only on other steps of the process, on DS intermediate, final bulk, and also to monitor the stability of the product at DP and at drug substance. For new product, and it's true for VRVG, the clinical trial design to generate the clinical data to support the potency criteria is key. As I mentioned, for VRVG, our acceptance criteria will be based on clinical data, meaning it's very important that the clinical development plan is developed accordingly, and also that so defining the dose we inject in human during clinical development is also very important, and it needs to be considered. We need to take into consideration, of course, process losses and also product stability, with the advantage of our rabies vaccine that it is very, very stable, and we hardly see any decrease during the shelf life. For our commercial product, the strategy we use to define our acceptance criteria is based on the consistency approach, and based on as I showed, based on a significant amount of data, more than 250 lots tested both in NIH and in ELISA, and that's where we were able to also to demonstrate that ELISA is much less variable as compared to NIH, and And also for VeroRab, we implemented ELISA not only on the drug product to evaluate potency, but also in intermediates for stability monitoring and also for process consistency monitoring. Next slide, and I think so. This is an arrangement of the main, not the only one, but the main people that contributed to this work. But it involved much more people within Sanofi. Thank you. Now it's for you, Eriko.
Thank you. Greetings to everybody. I'm Eriko Terrao from the Council of Europe EDQM. I'm a scientific project coordinator for the Biological Standardization Program, and I will take you through, to the second part of our presentation, explaining how we are moving towards a global harmonized change for human rabies vaccines to in vitro potency for in vitro potency testing. I will just disappear behind my slides. Thank you, Laura. Despite there have been several international initiatives for the development of an alternative in vitro method for the potency control of human rabies vaccines, and they all conclude on the feasibility of an ELISA approach. However, despite the development of various alternative approaches, the global acceptance for the replacement of the NIH test by an ELISA test is hindered not only by the fear factor that Dean and other speakers mentioned before, but also by the absence of a common standardized method. Next slide, please. So, why establishing a common standardized replacement method is interesting? Well, of course, if you have an acceptance at large, ideally at global level, it means that laboratories do not need to maintain multiple validated method for lot release testing, which means that the operators can focus on one method and they can gain also increased proficiency, which means also that they have a higher precision, not only because the ELISA approach is much more precise than the in vivo testing. The in vivo or the ELISA approach has also shorter lead times than an in vivo testing, which means all this means that the resources at the level of the lab are optimized and time is money, so it is much more cost effective. So now, to have a common standardized replacement method, what are the prerequisites? First one is that there's no proprietary rights on the method, so the method should be accessible to all users. The method also needs to use reagents and equipments that are accessible. It should not be rocket science. And more difficult to evaluate for one isolated laboratory is that the method should be applicable to most and ideally to all marketed products and be transferable and a robust method. And for these two main points, you often need an international collaborative project that would generate data to fuel the international initiative towards global change. Next slide, please. So for the human rabies vaccines, 3 steps were identified. The first one is to know what methods are available and which one to select. The step 2 was to check whether the selected method is suitable for global use, meaning it is transferable to all laboratories worldwide, and also that the method is applicable for routine release testing of most of the products. And with this, the data from these two first steps, you can move to the third step, which is the international agreement and implementation for the replacement of method. Next slide, please. So the first step was taken on board by the EPA project. Next slide, please. Yeah, So EPA stands for the European Partnership for Alternative Approaches to Animal Testing. It is a collaboration between the European Commission and the and industry stakeholders, which aim at promoting the use of the 3 Rs for meeting regulatory requirements through better and more predictive science. So the whole organization is under a steering committee. There's also an advisory body, including representatives from the civil society that are involved in this EPAA, and the secretariat is located in Brussels. Next slide, please. So, yeah, the EPAA in 2012 organized a workshop in Arcachon. That's what we commonly call the Arcachon 1 meeting. And during that meeting, an international working group, including public laboratories and manufacturers from all over the world, as well as academia, WHO, and EDQM, was created. The group was coordinated by Jean-Michel Chapsal, and the group made actually an inventory of available sandwich ELISA that were pre-validated. And these ELISA methods had to use well-characterized monoclonal antibodies as much as possible, recognizing in the case of human rabies vaccine, it is very specific, the protective trimeric form of the rabies glycoprotein, because that's the most important antigenic site. So the working group launched an inter-laboratory study to select the most appropriate ELISA method. Next slide, please. Yes, so 5 laboratories were involved in this study. They tested 3 ELISA methods, and they tested 3 different products covering 3 virus strains, 7 samples, including intact, heat-degraded, and a mix of spike-degraded products were tested, and they used the WHOIS as a reference standard to express results in IU. And the study, you can see the results at the bottom of this slide. So you're going to have a slide. The study identified one method to be the most discriminative, and it is the one here in purple that uses 2 antibodies, monoclonal antibodies, the 1112 and D1 clones. Next slide, please. So after the analysis of all the results, the EPA organized a second workshop in 2015, so 3 years later after the first meeting, and the working group determined that the GP ELISA, so the method B from the former slide, which was developed by Sanofi Vaccines, was the most promising method for further evaluation as a global harmonized method. So we checked, of course, that the method, there was no proprietary rights imposed by the developer. It used highly characterized specific monoclonal antibodies that were owned by public laboratories, meaning that they were accessible to all interested laboratories. The ELISA recognized at least 3 virus strains used for vaccine production, and that was the data from 2015. And since then, you will see in the next slides that actually at least 6 strains are recognized by this ELISA. And also the EPA study showed that these preliminary data support good transferability of the method. So all the results from this EPA study were published in a peer-reviewed scientific journal, Vaccine, and you can see the reference here. And all this allowed to move to the next step. Thank you. So the next step is the check whether the selected method is suitable for global use, and that was done under a project BSP 148. Next slide, please. Yes, so the BSP stands actually for Biological Standardization Program. It is a program co-funded by the Council of Europe, EDQM, and the Commission of the European Union. It was set up in 1991, actually, to organize international collaborative studies for the establishment of common reference standards and critical reagents. So to be used as distributed as official standards, but also for the evaluation of transferability and robustness of common, either new or improved testing methods to be used in official texts, such as mainly the European Pharmacopoeia monographs. So because the steering committee includes members from the group of experts from the European Pharmacopoeia, but also members, representatives from the EU Commission, the European Medicines Agency, but also WHO. The BSP program is actually independent and is ideal because it holds discussions with all interested parties worldwide, not only Europe, but wider. It works for the improvement of international harmonization, because we organize studies not only for Europe, but also with other organizations outside Europe. And we ensure a link to official texts such as the European Pharmacopoeia text. Yeah, next slide, please. So further to the EPA study, in 2016, we launched the joint EDQM BSP EPA project, called it BSP 148, and we have for project leaders Sylvie Mongeau from the French agency and Jean-Michel Chapsal, who was in charge of the EPA study, and I am the scientific coordinator. The BSP 148 project is divided in 3 phases: the preparatory phase, a collaborative study, and then a third phase, which is a reporting study. Next slide, please. So during the preparatory phase, we actually fostered the establishment of agreements, licensing agreement between the owner institute of the antibodies with not one, but 2 commercial suppliers. And this took most of the time, actually, almost 4 years. During that time, we also procured test samples for the collaborative study from 7 manufacturers worldwide. 11 samples were procured, covering 5 virus strains, and these samples had various potencies. All these samples were pre-tested by 2 reference laboratories to determine the pre-dilution, and we qualified also lots of critical reagents that were standardized in the phase 2. We also determined the statistical data analysis models so that we could elaborate a detailed SOP, study design, and study protocol. Next slide, please. Yeah, so this is just a quick slide showing how we determine the statistical analysis model for the data analysis. This was really important. We asked laboratories to perform a full dilution dose response curve, including 12 points, which is quite unusual. And our statistician actually managed to fit 2 statistical models to this second dose response curve. She had identified the 5PL model and the parallel line model. And for the collaborative study, we actually selected 8 points out of the 12, and we optimized all pre-dilutions of the sample so that the linear range and the upper and lower points of the dilution curves were covered. Next slide, please. So the phase 2 we had 31 laboratories who enrolled in that study. These were public or national control laboratories as well as manufacturers from all over the world. And they tested a common set of 11 marketed vaccines using the IS as reference standard. And all laboratories used a common SOP with standardized critical reagents. And all data were centrally analyzed at EDGAM by our biostatistician. Next slide, please. So the phase 2 was launched in December 2020 with the dispatch of the samples. During the whole year to 2021, we provided technical support to all participants to ensure method transfer. The analysis, the data, all the 25 laboratories reported results by February 22, and all the central data analysis and the report was finalized by April 2023. So it must be noted that due to the limited availability of the samples, also the limited study timeline that we could allow, and also the pandemic context, some reported data were generated from suboptimal assays. But the next slide shows you an overview of the results for the 11 samples. And you can see that the different colors actually show the potencies calculated by using different models, so the 5PL, using all subsets of data or only the subsets of data coming from assays that were conforming at best to the SOP. And you can see that all the results are quite similar. Next slide, please. Yeah, so this slide summarizes the conclusion of this study. So the collaborative study confirmed that the method is applicable to all tested strains, so all 5, and actually one participant tested its own product, which is of a 6th strain. So, and it worked. So the potency estimates were similar between participants and centrally calculated values, meaning that this is not rocket science for the calculation. The assay precision was satisfactory despite the suboptimal method transfer. So all confidence limits of the assays were within 80%, 125%, which is much, much better than the 400% reached by the NIH test. The assay repeatability was satisfactory, so the intra-laboratory variation was good, with GCV being lower than 15% for most laboratories and even as low as 1-2% in some laboratories. And it must be noted that the largest variation were observed for laboratories optimizing testing conditions in between reported assays, and that somehow artificially increased the variation calculated here. As for the assay reproducibility, so the inter-laboratory variation, it was very much satisfactory. Again, despite the suboptimal method transfer, the inter-laboratory variation of the G means was about between 6% and 13%, depending on the sample. And actually, a higher variation was observed for some samples that required higher pre-dilution, which means that the variation in this case was linked to the efficiency of method transfer and also the proficiency of the operators. Next slide, please. So with these good results from the phase 2, we launched the phase 3 in last December. It is the reporting phase, and this phase looks into the applicability of the GP ELISA to routine batch testing. So it is a kind of a simulation of a real-life situation where participants are requested to test as many batches of different products as possible with the standardized ELISA in order to generate data that could support discussions on future specification, say validity criteria for in the official texts. So for this phase, we have between 19 and 25 laboratories. We have actually 4 additional new participants since phase 2. Again, from public and manufacturers from all regions, and all these laboratories should have access to routine batches of marketed vaccines, because we are not providing samples to the participants. And of course, these laboratories have must have fully transferred the ELISA. So the data reporting for this phase is awaited by December this year, and all the data will be centrally again analyzed at TDPM before preparation of the report. Of course, at the end of the phase 3, a publication will be issued in a peer-reviewed scientific journal, and we will organize with EPA a symposium for discussion on method implementation, so that the proposal for the global replacement of the in vivo potency test by a standardized ELISA can be proposed, and that will be hopefully in 2025. And the last slide is to thank all study participants, and also to you to show how global this initiative is. Thank you for your attention.
Thank you, Patrice. Thank you, Enrico. Give me 1 second so we can go to see.
I started to answer some question in the chat.
Thank you. Thank you.
I just want to summarize what I wrote in the chat so that anybody can hear it. So I just want to mention, and I didn't say it, but I want to say that Sanofi did not issue any patent for the assay, meaning anybody can use the assay. It's published, so anybody can use it. And in addition to that, the 2 monoclonal antibodies for the ELISA is now available commercially, so anybody can purchase the monoclonal and perform the assay, so it's free of use, no restriction on that. The other 2 questions I got and I answered in the chat is that, is there any authority that approved an NIH test replacement by ELISA yet? The answer is yes. For our VeroRab vaccine, we submitted a variation for the replacement of NIH by ELISA, and we got the approval for EMA. We got the approval of additional maybe 2 or 3 countries, but I don't recall exactly because I'm not completely in the field, but I know that other international submissions are ongoing for VeroRab, and our expectation is that to have this variation accepted in the coming month for VeroRab. So we are hopeful on that, of course, but we got the positive feedback from EMA, which was a very good start. And the first question I answer is that the question whether we can set a statistical correlation between ELISA and the in vivo NIH test. And I just reply, as mentioned by Dean, and others can comment if they want, it is almost statistically impossible to generate a statistical correlation between an in vivo assay, which is very variable, and an in vitro assay, which is much less variable. So it is almost impossible. So, and this is what is mentioned in the European chapter 5 to 14 for in vivo assay replacement by in vitro, saying that if there is an expectation to have this statistical relationship between a variable in vivo and of an in vitro assay, that would be almost impossible to replace. So we should not foresee, or we should not, I would say, look for a statistical correlation, because, as I said, it's almost impossible to achieve. Go ahead, Enrico.
Yes, just one point to highlight. In the case of the GP ELISA, what is really important also is that despite the fact that there's no statistical correlation that can be established, Sanofi performed assays using clinical lots of different potencies, and actually you showed that the ELISA can discriminate the clinical lots with different potencies, and that's really powerful as an information.
Yeah, that's what I mentioned, the suitability package we generated. So we generated a suitability package to demonstrate the performance of the assay, but the statistical correlation per se between the two assays, as I mentioned, as I replied in the chat, is almost impossible and should not be expected for in vivo assay replacement.
Thank you, Patrice, and thank you, Enrico.
Thank you.
Thank you for all the answers that you provided already on the chat. So I keep inviting people to do comment. And thank you for the collaboration. Without further ado, let's move to our next very important case study that is actually explaining what is the current strategy that the European Pharmacopoeia is implementing to replace the rabbit parogenicity test. And our speaker is Dr. Emmanuel Cherton, that is going to present all the strategy and work that DQM has put in place since the last few years to make this important transformation. Thank you, Emmanuel. The floor is yours.
Thank you very much, Laura, and thanks for inviting me to this event. Can you hear me well?
Yes, Emmanuel, yes.
Yes.
You can see you and hear you very well.
Good. So I'm going to present you the Pharmacopoeia strategy for phasing out the rabbit parogen test. In such presentation, I would normally start by explaining what the Pharmacopoeia is, and so I will spare these slides and just would like to indicate to you that recently we carried out a training on biologicals, which starts with a general concept module, and you may click to this link and listen to these things if you are interested. There were 4 modules, one general one, on general concept 1, followed by biotherapeutic and ATMPs, and then one on plasma-derived products and vaccines for human use, a module on reference standards, and a module on microbiology, including parogenicity. So I dive in directly to the parogenicity topic, and presenting the 4 tests that are available in the Pharmacopoeia to address parogenicity. First of all, the rabbit parogen test, which exists since 1971, so more than 50 years ago. Then the bacterial endotoxin test, which relies on the substrate extracted from horseshoe crab. An in vitro parogen test called the monocyte activation test. And last but not least, the bacterial endotoxin test using recombinant factor C, which then avoids using a reagent coming from animal species. So despite the fact that all these tests are available, and the animal test still remains to be widely used across Europe. So it's been decided together with the experts of the Pharmacopoeia and a joint initiative with European Partnership for Alternative Approaches to Animal Testing, EPAA, to get rid of the parogen test. And this will go with the elaboration of a new chapter 5113, parogenicity. So I will explain what this is, but you can all read the detailed strategy in a paper which is available on the EDQM website. So there are 60 texts which refer to the animal test, and these texts are of different categories. You have general monographs. Substances for pharmaceutical use is the one which has the largest impact. Dosage form monographs and the parenteral preparation monograph is the one which has the largest impact. We have a number of individual monographs. So for you today, it's important to know that there are 17 vaccine for human use monographs referring to the rabbit parogen test, and also a number of general chapters. So the idea is that the strategy will be as explained on this slide, and this strategy was approved in 2022. So that at the moment there are 60 texts which refer to the rabbit paragen test, but in the future it will be up to the user to decide how they want to address paragenicity strategy, and this will all be explained in chapter 5.1.13. So based on risk analysis and the potential presence of non-endotoxin paragens, based on the stage of manufacturing process, the user might choose to do either a test for bacterial endotoxin or a MAT, or to do both. And then they will also decide on the testing strategy and on the limits. And this whole strategy will lead to in the 12th edition of the Pharmacopoeia, to the inclusion of a new general chapter and the deletion of the rabbit paragen test from all these texts. So I would like to show you examples. For example, in the general monograph Substances for Pharmaceutical Use, at the moment, there is a test for BET and a test for paragens. So these two tests are going to be removed and replaced by the reference to the new chapter. And for example, this is a typical individual monograph which refers to the rabbit paragen test. So the plan is to delete the paragen test, and the fact that the general monograph Substances for Pharmaceutical Use applies to all substances, this specific monograph then will have to comply with the general monograph. So you will see no animal test anymore. The general monograph 2034 will apply. Another example on a general monograph, which is very important, is the parenteral preparation monograph. The BET test, pyrogen test, will also be replaced by a reference to the new chapter, and the rabbit paragen test will disappear. This is an example in the field of blood products, so I will skip that. Same principle, just to spend a little bit more time, because you have a vaccine profile today, so just to spend more time on the different individual vaccine monographs which refer to the rabbit paragen test. So it's been a very complicated exercise, because this monograph refer to rabbit paragen test at different stages of the manufacturing process. So the expert responsible, group 15, responsible for this monograph, have had to look really very carefully, case by case, on what to do with these references. So all these texts are being revised, and but most importantly, the general monograph Vaccines for Human Use, monograph 0153, will also be revised, and this is explained on this slide. So there will be a new paragraph which in the production section, in the general provision part, which will say that paragenicity is characterized during development studies and controlled whenever revalidation is necessary. And the new general chapter 5.1.13 will be referred to. And in the test section, the BET test will be replaced by a paragenicity test, which once again refers to 5.1.13. And the consequence for this inclusion of the paragenicity test in this general monograph will be that in all individual monographs on vaccines for human use, the paragen test will be deleted, and the requirements of this general monograph will apply. So no more rabbit paragen test in these individual monographs either. So, I just wanted to show you the explanatory notes for all the revised texts. So these revised texts were published all for proposed revision in Pharm Europa January of last year. And the notes explain that the exercise will ultimately lead to the suppression of General Chapter 268, and that the manufacturers still using rabbits are very much encouraged to find other solutions, non-animal, which are non-animal based. But importantly, also, the revision of this text does not call into question strategies involving the test for bacterial endotoxin that are already used by manufacturers to control pyrogenicity. So it means that if somebody has already replaced the rabbit pyrogen test by a BET, they will not be affected by this whole exercise. So this is just a slide which shows you that we have largely advertised this project, and today I'm advertising this to you. But it started all in June 2021 when the project started, and then there were lots of workshops, either PDA, Pharma Lab, NC3R most importantly because they had an impact on WHO guidelines, and WHO guidelines are going also to be revised, but in another context. But we also intervened there. There were some microbiology conferences. The 11th edition conference at EDQM, we made a lot of publicity to this project. And last but not least, in an EPAA EDQM international conference that took place last February of 2023, to also discuss that very largely within Europe and also with external, out of Europe stakeholders, because it is felt that this exercise would be very much beneficial if it was not only a European exercise. So this is the information about this event that took place in February. The main conclusion of the event, which encompassed 250 participants from all regions. So the take-home message was that in Europe, stakeholders are showing great enthusiasm towards the European pharmacopoeia strategy aimed at phasing out the rabbit pyrogen test. Outside Europe, the strategy is generally seen very positively. However, alternative methods such as the MAT are not described in detail in other pharmacopoeias, where the rabbit pyrogen test is still required in monographs. So the journey towards a complete removal might therefore take longer. However, that being said, there is international convergence toward the same goal, and so the spirit of all the stakeholders outside Europe is definitely for getting use of the animal test. What also could be concluded from this important event is that implementing the MAT has been greatly facilitated in the last years by the standardization of reagents, and it was very pleasing for us to realize that the simple fact that we announced that we were going to get rid of the rabbit pyrogen test triggered the creation of some startup or some companies who decided to develop MAT kits to be able to provide some available reagent when rabbits would not be available anymore for pharmacopoeia pyrogenicity testing. So the time has definitely come to switch from in vivo rabbit pyrogen test to in vitro. You can find all the slides, video, and training resources on this link related to this event. And also there is a paper which was written after the event which summarizes all the discussion, and it's a free paper that you can download also. So what are the next steps? Not many more left after all the work on all the text of the monographs which were published in Pharm Europa. And we are going to submit them all to the Commission in June, so the European Pharmacopoeia Commission. And if everything goes well, the date of implementation of this change will be July of 2025. So the publication of monographs without the rabbit pyrogen test. knowing that the MAT revised chapter is already going to be implemented this year in July. And our plan is also to have a complete deletion of chapter 268 from the Pharmacopoeia in January of 2026. So the project is on track for the deletion of the rabbit paragen test from the Pharmacopoeia in 2025. So bye-bye the rabbit paragen test. And I would like to acknowledge the tremendous work of the Bacterial Endotoxin Working Party of the European Pharmacopoeia and its chair, Dr. Ingo Spreitzer, who was present at all the discussions in all groups. So the texts in question are under the remit of a number of groups of experts. These are all cited there. So it was a tremendous work within all these groups and their respective chairs, and my colleague Gwenaëlle, who coordinated and who is still coordinating this project, is gratefully acknowledged. Thank you very much for your attention.
Thank you very much, Emanuele. Congratulations to you all for these important achievements and to be very close to this important transformation. Let me see if we have questions in the chat. It doesn't look like, but I feel that we can take out from your presentation is that science should lead all the transformations. The stakeholder engagement and the discussion with all parties is fundamental in order to find the right way to proceed. And I really hope that many other stakeholders in other countries can really get inspired by the work that has been done, taking care, of course, by the different complexity of the different countries and regions to deal with different type of products and different type of regulations. So do you have any general recommendations on how to start this process?
So I'm sorry, but the sound is not very good, so I don't hear you very well, but is the question about the process on how to establish removal of the rabbit paragen test outside Europe?
Yes, if you have any recommendations about some key principles on how to start it. Thank you.
Yeah, I think the first step would be to encourage the use of MAT, monocyte activation test, because there are lots of regions where this test is not even included in pharmacopoeias. So I think that's a very first. I'm just citing the United States Pharmacopoeia, who does not have such test, for example. So if there are some major pharmacopoeias who have a very big impact worldwide. So if they were to delete the rabbit paragen test, the very first step would be to make sure that there is an in vitro test which is available in the same pharmacopoeia. So for me, to promote the monocyte activation test is the way forward, and we've done that for many years. So and then the rest is maybe mostly political, and in Europe, we work hand in hand with the EU, and I mean the fact that the EU had this directive on the you know, protection of animals was also a trigger for us to react. So the regulatory authorities must also be just as convinced as European regulatory authorities to go this path forward. But when you think about a 50-year-old animal test, yes, for which you scientifically speaking, there is no, you know, no validation behind these tests, no suitability. You don't check whether, you cannot check whether a rabbit is suitable for the test. You just take a rabbit, that's it. And when you think about it, it's just an anomaly to be still using this animal to measure fever. So, yeah. So we keep hope that it's going to be removed from the planet in the years to come.
Thank you, Emanuele. One last question, which I believe is very important, because it explains exactly what you were saying, that we need to promote more the monocyte activation test. And the question is, if the MIT can replace both the rabbit parogenicity and the bacterial endotoxin test.
Yeah, I can see in the chat this question. So the monocyte activation test is an in vitro replacement of the parogen test. So it means that it can detect both endotoxin and non-endotoxin parogens. The BET test is a specific test. It's a bacterial endotoxin test. It measures only one category of paragenic substances. So the question, can MAT replace BET, for me is a sort of not very logic. So I would like to say the MAT, yes, the MAT is the in vitro replacement for the rabbit parogen test. And then why do other pharmacopoeias not describe MAT? I think you have to ask the pharmacopoeias. I cannot reply for them.
Thank you very much, Emanuela. Very appreciated. We move to our next speaker again in the interest of time. So thank you very much for
thank you.
Your presentation. And we continue with the representative from the European Pharmacopoeia. We have Laurent Mallet with us that is actually going to present another important advancement in terms of transitioning from in vivo to in vitro. Is going to cover the next generation sequencing opportunities. And thank you very much, Laurent, for being with us today. The floor is yours. Thank you.
Yes, thank you very much, Laura, and thank you, Emmanuel. I will not also present the DQM institutional slides, and I think you have seen that Emmanuel provided some links to have more explanation on that. So thank you very much for the invitation and for having various representatives from EDQM and also members of experts group to share the progress we have made on implementing non-animal testing. I hope you hear me correctly.
Yes, Laurent. Yes, we can.
Okay.
Thank you.
Okay. So I will give you a presentation on next generation sequencing for adventitious viruses, covering the ongoing initiatives and also some upcoming events. Particularly describing briefly what is next generation sequencing and how we have made some evolutions at the level of the European Pharmacopoeia, perspective on these NGS and HTS, and elaboration of a new chapter, which was already mentioned in the slides from Dean. An update on the ICH Q5A Revision 2 guideline with a focus on NGS introduction to replace in vivo assays, and a few information about upcoming events on NGS and some key references. So what is next generation sequencing? It's also called high throughput sequencing or massive parallel sequencing. It's the sequencing of acid nucleics with high throughput, scalability, and speed. You have different technologies which are available. I will not give the commercial names, but we have short reads, long reads. They can read the length. They have read length from hundreds of nucleotides to more than 50 plus kb. And one of the applications that can be done with this technology is the detection and identification of viral, extraneous, or adventitious agents. We use both terminology with quite a good sensitivity, but particularly an excellent breadth of detection, because there is no bias in the way the technology is able to detect these extraneous adventitious agents, with the capability to detect both known and unknown viruses. I will not speak about other applications which were mentioned in the slides from Dean, such as molecular consistency, which is also another application of NGS. So what about the evolution of the European Pharmacopoeia, and particularly why? The drivers for the change were particularly in 2010, but the NGS technology already started before, the contamination of a rotavirus vaccine by porcine sarcovirus, which was not detected by existing methods such as in vivo and in vitro tests for adventitious agents. It was published by Victoria and Al in the Journal of Virology, and the results showed that the presence of PCV1 viral sequences were present using new high-throughput molecular biology method, massive parallel sequences. So there was an emergence at that time of broad molecular methods for extraneous agent detection with degenerate PCR, family PCR, but also these NGS or MPS or HTS technology. There was a revision at that time of the WHO TRS on cell substrates, which I mentioned here, where there was a risk assessment strategy which was introduced, and as well as new methodologies which were really emerging at that time in 2010. There was also a need for convergence with FDA guidance for industry on testing methodologies. And in Europe, as mentioned by many of the speakers today, there was also the 3Rs context with the European Convention for the Protection of Vertebrate Animals used for experimental and other scientific purpose with this EU directive. We conducted at the level of EDQM a survey with vaccines manufacturers and CRO regarding contamination cases over a period of 10 years. And to be honest, there were only a few, there was no real contamination cases, and certainly not detected by in vivo methods. And there were a number of publications which were highlighting the gaps in compendial tests, where there was really a poor sensitivity of in vivo methods, which highlighted again the gaps in the testing packages, which was already visible when you think about the fact that some contaminants were not detected by the existing methods. So there is this article from Gombold, which was published in 2014, and an additional article from Rebecca Chitz and Paul Duncan in 2015. So there was a need to update the European Pharmacopoeia requirements at that time. So it was done by Group 15, already mentioned by Dean at the European Pharmacopoeia level, with 3 chapters. The chapter 5 to 3, where the tests for specific viruses such as NAT, PCR were introduced, but really importantly as well the tests for virusing using broad molecular methods such as HTS. As alternative to in vivo and specific NAT, or in addition or as an alternative to in vitro cell culture. So it was done in 2017 for both the chapter 5 to 3, which is dealing with cell substrates, and the chapter 2, 6, 16, which is dealing with viral seed lots and harvest in viral vaccines, but also covering the gene therapy products, vectors particularly. In parallel, as mentioned, and it was presented exhaustively at the beginning of the webinar, the chapter 5 to 14 was published in 2017, and there is a consideration for the substitution of in vivo method by broad molecular methods in this chapter. Perspective and elaboration of a new European Pharmacopoeia chapter. So, as you have seen, since 2017, these 2 chapters, general chapter 5 to 3 and 2, 6, 16, mention the HTS and foresee its use as part of the testing strategy for extraneous agents. However, up to now, there was no description of these methods or any guidance for their validation was provided. It's true for the European Pharmacopoeia, but also for other regulatory documents. The availability of these regulatory standards, including validation guidelines in the European Pharmacopoeia, was really seen as a very important point as a reference for regulators and manufacturers, while at the same time, there was a revision of the ICH Q5A guideline, and this revision was adopted in November 2030, and I will go to that a little bit later. And at the same time, a first panel of model viruses was developed by FDA and was approved by the WHO. There is a new one which has been approved in March with 7 viruses, as would WHO International Reference Panel for HTS. So a new chapter has been drafted by the European Pharmacopoeia Working Party, the HTS Working Party. It's called high-throughput sequencing for the detection of extraneous agents, 2641. It's a non-binding general chapter. It describes the technology, the methods, and all the aspect of HTS workflow. And it's also described guidelines for validation of HTS methods. It was elaborated by an international group of regulators, including also OMCL and industry from Europe, US, and Canada. And I am pleased to tell you that due to the work of this working party and my colleagues from the European Pharmacopoeia department, the draft chapter has now been published very recently and last Friday for public consultation in PharmEuropa. So I put the link here and you will discover the chapter because the slides will be shared. And the deadline for comments is by the end of June 2024. So please do not hesitate to comment this new chapter. This draft chapter describes HTS methodologies for the detection of viral extraneous agents in biological products. It includes all possible applications, meaning vaccines from both human and veterinary field, recombinant proteins, viral vectors used for gene therapy, and cell-based preparation for cell therapy. It really outlines the different steps of the HTS workflow, the design of the method, the analysis approach, the control used in the routine tests, and also the follow-up investigations. It also provides guidelines for HTS method validation, including recommendations for the selection of spiking material for validation at the manufacturer level, and the evaluation of the relevant performance characteristic for HTS. I will switch to an update on the ICH Q5A R2 guideline focused on NGS introduction. So this ICH Q5A guideline has been adopted on the 1st of November 2030, and I put here a link to the approved version. There is an extension of the scope of the guideline. It's important to mention that because now it's defined as products that are amenable to viral clearance without negative impact on the product. So as a consequence, it includes some genetically engineered viral vectors and viral vector-derived products. So it includes, for example, virus-like particles, protein subunits, and nanoparticle-based protein vaccines. So in this introduction, in this new guideline, revised guideline, there is an introduction quite important, which is the introduction of the next generation sequencing for virus detections with, if we focus on in vivo tests, specific opportunity to replace existing methods with targeted or broad, what we call also non-targeted NGS, which are highlighted. And it's particularly the case for antibody production tests, in vivo assay, as well as for in vitro assays and nucleic acid techniques. I have extracted some sentences from this ICH Q5A guideline with a focus on in vivo assay, particularly where it is clearly written that you can replace non-targeted NGS as a replacement for in vivo assay. Also for the antibody production test, which are done particularly for products which are manufactured on rodent cells or where you have some rodent cell in the history of the manufacturing. So it's called the MAP-HAP test. Here there is an encouragement to switch to either PCR or targeted and non-targeted NGS. Why targeted and non-targeted? Because in fact, these tests detect a selection of viruses, these antibody production tests. So you can design a targeted NGS approach to replace these animal assays by NGS. And then regarding the next generation sequencing chapter, you have also sentences clarifying that you can use non-targeted NGS, meaning the broad approach, to replace the in vivo test with broad virus detection of for unknown and unexpected viruses species. Without to-head comparison, what does it mean? It means that you don't have to show exactly a comparison with the existing method, meaning that you can rely on existing published data, particularly if they have been generated with available references. And it's really aligned with the spirit of the chapter 5 to 14 from the European Pharmacopoeia, which has been presented earlier. I will switch quickly to the upcoming events. I have tried to give you an idea of which events are coming that could be of interest if you want to be to familiarize yourself with NGS, in addition to all the documents that I already mentioned. There is an IBS DCVMN meeting, which is about NGS testing to replace in vivo adventitious testing for old and new vaccines. It's on invitation only. It's for DCVMN members. There is a PDA virus conference, Viral Safety Reloaded, with the finalized ICHQ5A R2, which will be particularly the focus of the conference in the session. And there is also an NGS session. There is also a workshop, a dedicated workshop associated to this conference on the 28th of June. And there is also a pre-conference workshop from the ECA European Microbiology Conference that will focus on next generation sequencing on the 25th of June. So as you can see, it's quite busy at the end of June if you want to learn some information about NGS. The next one is the 4th IABS conference on next generation sequencing for adventitious virus detection in biologics for humans and animal detection. So it's really the conference, the 4th one. It's a big conference focused on NGS. I will have the pleasure to co-chair this conference with Arifa Khan from the US FDA. It's in December in Frankfurt, Germany. You will have on the 3rd of December a one-day dedicated NGS training workshop that will go step by step on the different phases of an NGS technology and what you need to know about these different technologies, including long read and short read. And really the goal of this conference is to bring together academia, technology providers, representatives from industry, and of course, regulators, regulatory bodies. So this is, you have the link here, and you can have access also to the call for abstract, which has been published very recently, that you can have a look, and also the registration form. I have here listed some key references, including the reports of the previous conferences for the IABS conferences, which were really published in Biologicals and which are really exhaustive regarding the content of these previous conferences, the ICHQ5A, the draft chapter again, and also an example of validation. And I would like to take the opportunity to thank all the contributors to all the different topics I have covered in this presentation, including the EDQM Group 15 and its chair, Svein, who will speak just after, the EDQM HTS Working Party and its chair, Dr. Johannes Blumel, who is also involved in the ICHQ5A from PI, Gwenaël Thuy and Emmanuel from EDQM, because they have been extremely supportive of all these sequencing, next generation sequencing and European Pharmacopoeia revision. They are in charge, in fact, of this activity. And the ICHQ5A expert working group, as well as Dr. Arifa Khan, who is extremely involved in this NGS development and implementation. Thank you very much for your attention.
Thank you very much, Laurent. I don't really see questions right now in the chat or in the Q&A. I really wish to thank you for sharing all the references and the information about the upcoming events. I think that there is really a lot of opportunities to better learn how to implement the NGS for the adventitious viruses, and hopefully this is really going to lead again together with the ICH Q5R2 into global harmonization and the implementation of this technology. And we are glad that all these activities are ongoing and you reported about that. Thank you very much, Laurent.
You're welcome.
I would like to move to our last case study. We go back to a very important group of vaccines. That is again diphtheria, tetanus, and pertussis with Svein Rune Andersen, who is a representative from the Norwegian Institute of Public Health and also the chair as Laurent said, of the DQM Working Group 15, and is really going to go through all the opportunities that are moving us towards the end of the in vivo toxicity testing. And hopefully you will learn about the opportunities to delete obsolete tests. Thank you, Sven, for being with us. The floor is yours, and thank you very much again.
Thank you very much. I'll just share my slides. Can you see my slides now?
We can. They are not yet in the presentation mode.
Okay, let's see. Bear with me.
Yeah, don't worry, Sven.
Okay, there we are. Okay. Yeah, thank you very much for being invited to this meeting. I'm going to talk about the title is towards the end of in vivo toxicity testing, and we are on a good path, but maybe I should have added a question mark at the end. You will understand why later. So in Group 15 that you heard a lot about recently, there is we have reduced toxicity testing in animals, and we have covered the diphtheria vaccine, the tetanus vaccine, and acellular pertussis with regards to pertussis toxin. I also talk a little bit about the abnormal toxicity test, because even if Dean and others have covered that previously. But in our struggle to review all these requirements for toxicity testing, we have taken a scientific approach, and Dean talked a lot about that. We have been reviewing and rationalizing the strategies, and this has been based on accumulated information and collaborative studies. So I think considerations prior to deleting a toxicity test, or any test for that matter, and I think it's important for the European Pharmacopoeia, which is part of the regulatory legal framework for Europe, to be up to date and have relative or relevant requirements. So I think you should always ask the question, is the test suitable for its intended use? Is there an alternative test that is equally or more sensitive or reliable? Is the test relevant for today's manufacturing requirement? And the introduction of GMP, and also now more recently the consistency approach, we need to keep that in mind. Is the test needed considering the overall strategy? And that's linked to the question about, of course. And finally, what does historic testing results from routine testing or vaccine development tells us about the relevance of the test or the need for the test? So just very quickly on the abnormal toxicity test, or the general safety test, as it's called in the in US, or innocuity tests in WHO guidance. This was developed early in the 1900s to detect toxic level of phenol and preservative in serum products. And I think, if I'm not mistaken, I think the requirements for the test was to use one mouse. Obviously not fantastic, but it could differentiate between two high levels of phenol if the mouse died. And also it's been used for extraneous contaminants, so tetanus toxin and spores in sera, and for that guinea pig was used. So essentially these two tests were combined in the general safety test and have been unchanged since. And retrospective analysis of data has shown that this test is neither specific, reproducible, reliable, nor suitable for its intended use. And there are several publications on this. I cited Ducot et al. From 1994, but there is also a very good paper from at PI that Joris mentioned previously, which is worth taking a look at. But because of this publications, the test was deleted in 1998 in more than 80 monographs in the European Pharmacopoeia. However, there was some reluctance in the then Group 15 to actually move it all together. So it was moved to what we call the production section. And this describes things that have to be done during development of the vaccine. So it's no longer a routine test, but it's something that you need to, is part of the characterization. But back in 2016, there was a workshop with global stakeholders organized by EPAA. And it was quite clear that from the discussions there that the ATT lacks scientific relevance and its omission does not compromise the safety of biologic. So it was a consensus to strive for deletion of this test from regulatory requirements globally. So eventually in 2017, the ATT was deleted from all monographs, including the vaccine monographs in the European Pharmacopoeia. And FDA or regulation, I think is a short form, deleted it actually slightly before us in Europe. And it's also gone from the European WHO guidance. So moving to ACell pertussis, Dean talked about this. There was this histamine sensitization test. Was established the CHO cell test, the clustering assay, which is far more sensitive than the HIST test. And also, as Dean mentioned, this CHO cell tests have been validated in collaborative study. There's a reference there to this paper in Pharma Europa Bio, which is worth taking a look at. And Group 15 then elaborated a chapter on residual pertussin toxin, which describes this test. We also, in this process, we looked at other ways, if there was a need to change, to make additional changes to the testing requirements. And we found that based on historic data, the pertussis toxoid is stable. So we didn't see any higher or any risk at all that this will revert to a toxin upon storage. And that leaves us with the option to only test the purified toxoid before it's mixed in the vaccine as a final product. But to somehow cover the for new vaccines, it's important to know that the detoxification process actually gives a stable toxoid, and that doesn't revert to toxin. So in the production section of the monograph for ACell pertussis, we introduced requirements that the detoxification process should be demonstrated to avoid reversion. So now there is only one in vitro test for pertussis toxin in the monograph, and that's performed on the purified toxoid before it mixed with the rest of the components in the vaccine. So moving on to diphtheria vaccines. The old version of the monograph had a test that was in the production section that called the specific toxicity test, where one was expected to inject subcutaneously 5 times the single human dose into 5 healthy guinea pigs. And if they died within diphtheria toxemia, then the vaccine didn't comply with the test. However, and there was a routine test for diphtheria toxin called the absence of toxin and irreversibility of toxoid. This is cell-based and it's the Verocell test. And it has a very good sensitivity. It's an easy readout. You can actually use the pH indicator in your medium in the cell culture as the readout. And this test have the 2 elements of absence of toxin and irreversibility of toxoid to toxin. And this is simply done by incubating the vaccine at either 2 to 8 degrees or at 37 degrees for 6 weeks and then test. So since the cell-based routine vaccine is more sensitive and reliable than the animal test, we didn't see it as a great risk to delete the specific toxicity test. So moving on to tetanus, it has the same kind of specific toxicity test. It's a slightly shorter observation time of the animals. And for the same reasons that we had for the diphtheria vaccines, namely that the routine testing of purified toxoid, the irreversibility of toxoid and absence of toxin, we decided to delete this test performed during development. And however, in this process, we realized that there were data available that showed that incubation at 37 degrees actually the tetanus toxin lost its neurotoxic activity. So it doesn't seem that to be very relevant, or obviously the test was not suitable for irreversibility. So the consequence of that was that the test has now been revised. So the irreversibility part of this test in guinea pigs have been deleted. As I said, this is a test in guinea pigs, so it's still an animal test, but it's more sensitive than the previous or the specific toxicity test. So since there was no point of maintaining the incubation at 37 degrees, we now are left with the absence of tetanus toxin at the level of the purified toxoid, which is when a potential toxin should be at its highest concentration. So, as I said, this is still an animal test, but there are in vitro tests in development, like the so-called barnacle assay. It is currently being evaluated in a collaborative study looking at both veterinary vaccines and human vaccines. And we will relatively soon in Group 15, together with our colleagues in the veterinary expert group, the Group 15V, discuss how we can open up for an alternative test, to the animal test in the pharmacopoeia. So I think that was more or less nearly what I had to say. I just want to conclude that by applying scientific approaches adapted to the specific vaccine, the European Pharmacopoeia has improved testing requirements for toxicity in vaccine monographs without jeopardizing the safety. So we are left with improved methods. We have heard several times that animal tests are difficult to perform. They are not always reliable, and there are alternative tests that is less variable and more sensitive. So that leaves us with a reliable testing strategies for toxin testing. And of course, through this exercise, we have achieved reduction, replacement, and refinement, which leads to less suffering of animals. So thank you for your attention, and I will just acknowledge Wenwell, who's the secretary in the secretariat for Group 15, and Dean Smith from Health Canada. But of course, the entire Group 15, which is a fantastic group of people and experts on vaccines, have contributed to this work. And with that, thank you.
Thank you, Svein, for your presentation and overview about everything that the Working Group 15 has been done in this specific context. I think that this is really going to show how important it is to apply, to use all the experience and expertise, especially on products that have been in the market since a lot of years. So you can really have a lot of strong data to really take this type of decisions. And I hope that this data-driven approach can be also taken up by other organizations worldwide and slowly taking those important decisions. So thank you, Svein. And again, if anyone has any question for Svein, please feel free to write in the chat. We still have a few minutes before we close this webinar. Thank you, Svein. And with this, I really want to introduce our final speaker, Robin Levis from the US FDA. She's actually going to help us to wrap up this webinar. Thank you, Robin, for being with us today. Your words will be very much appreciated. Svein, if I can just quickly check with you if you can close your sharing.
Ah, sorry.
Thank you. Don't worry. Thank you.
Am I gone now? Yeah.
Yes, everything is perfect. Thank you. Thank you, Svein. Thank you, Robin.
Hi, good morning and good afternoon and good evening to everyone. I really have the nice opportunity here to just wrap things up, and I would first like to thank Laura and her group who helped plan this webinar. It's really important to have these types of dialogues and public-facing interactions to really engage globally and to encourage these dialogues. So it's really wonderful to see such a broad participation in today's discussion. I really also want to recognize the speakers who really embody both the thought leaders and the action leaders for this really important initiative and set of work in terms of the establishment of science-based assays that will hopefully serve to replace and substitute the current animal-based release testing. And in fact, this is only a small representation of the global engagement and people involved in this. And so I would also like to acknowledge the NC3R's group, who has been really instrumental in an Gates and WHO funded initiative to work on the identification of lot release tests and quality control tests that are animal-based, that are still in the TRS and the guidelines, the WHO guidelines. And there's a large initiative underway to ultimately update the guidelines, but prior to that, to write a WHO a WHO document that will detail the current state of the art, as it were, with respect to replacement assays, and make a global statement, very much like the 5214 has, regarding the critical engagement that's required to get the best assays in place where possible. I just want to highlight, based on the presentations today, the successes that we can celebrate. First of all, thanks to Dean for his very nice introduction and description of the 5214 document from the European Pharmacopoeia and how that has impacted efforts in this line. I also want to highlight the VAC2VAC consortium, and that initiative was really, I was very humbled and proud to be part of that, those discussions in that organization, because it really was a model on how we can engage regulators, industry, academics, and really work towards getting some solutions to alternatives. Also, in terms of assay successes that we can celebrate, the removal of the ATT is very important. We need to get, much of the world has acknowledged that this test is no longer needed. And I think that in those regions where the test is still being performed, there is an awareness and a movement to remove that test. The removal of the histamine sensitization test, that's a relatively new endeavor, and hopefully that will also be taken up globally as an assay that's no longer required. I want to acknowledge the extensive work and near completion of the studies for the rabies ELISA development as a replacement test, and that for me was my entree into this field some 25 years ago. I took over working on the rabies virus vaccines at the FDA, and immediately was engaged in dialogues with respect to the development of a replacement test. And I'm really happy to hear and hope that before I retire in the next few years, this test and the study will be completed and we will have the licensed alternative assay. And finally, I want to acknowledge the work on the pyrogenicity assay and the development of the MAT and the other assays to replace both the rabbit test and the LAL test, the horseshoe crab-based LAL test. And kudos go to EDQM and the groups there who are working on updating the monographs and are really forcing the rest of the world to confront these really difficult questions. And finally, one of the other really major accomplishments that I think is going to move these discussions forward is the development and implementation of NGS as a means to characterize and qualify both substrates used in manufacturing as well as product quality itself, as Laurent very nicely described. And so just in conclusion of my comments, my brief comments, I just really want to say that back to Dean's kind of opening comments to us, that is really the best science drives the decision making and really makes it so that across all stakeholders we have confidence in the assays and really feel comfortable with the both development and implementation of these new assays. And using a science-based approach really is the only way to reach the goal of developing and implementing these alternatives. Again, to that end, I highlight the VAC2VAC initiative, and I really encourage everyone on here to consider this type of collaboration, whether it's regionally, like the VAC2VAC was Eurocentric, but really has implications globally, or globally. And of course, all of that depends on getting the appropriate funding. But I really, really encourage us to try to find space for those types of initiatives. And I think the NC3R's group in London is also very engaged in those types of activities. Finally, secondly, I want to thank the sponsors who have really been willing to engage and share across groups to facilitate getting the development of these assays. And I think to my regulatory colleagues globally, I say it's really important for us to be engaged and receptive to these discussions. We have to not only join the discussion, but also to the best of our ability, not just be responsive to what's happening, but actually facilitate the implementation of these assays. And so I think that's really a hard ask for us as regulators, but I think we can see from the successes that one of the reasons they were successes were that the regulators have been engaged in the dialogue. So with that, I will bring my comments to a close again. I really want to turn it back to Laura for any closing comments, but thank you again to the speakers, and I hope we get the opportunity for us to all get together and continue these dialogues on a global basis. Thank you.
Thank you very much, Robin, and thank you very much to all the speakers for joining this event. We really wish you to see online again during this year. We are going to have some other important webinars and workshops dedicated to the monocyte activation test implementation and the different regional complexities. And we will also want to bring up more discussion on the tetanus, diphtheria, and pertussis containing vaccines. So stay tuned on all our activities following us on the Animal Free Safety Assessment collaboration website and on LinkedIn. So thank you very much, and we really hope to continue to collaborate with you all globally for this important transformation. Thank you very much for being with us today. Goodbye.